Welcome to the second day of the GYNOCARE Conference, which will then be followed by the MC meeting. We're happy to welcome, apart from Dr. Federico Ortelli from Brussels, but even Dr. Aldona Gembalik, who joined us today, who is our administrative officer on behalf of COST. Okay. So basically, today we've got another lineup of interesting talks. And then after the second coffee break, there's the MC. But we'll see how we are with time. Maybe we can push that closer as well. Okay. All right. So I'll start with the first presenter, a very good colleague of mine who we've known for a long time, Dr. Miriam Azzopardi. Dr. Azzopardi, she's a consultant in public health medicine at the Directorate of Health Information and Research. She's the consultant in charge of the Maltese National Cancer Registry, as well as the National Rare Diseases Register. Prior to taking over these registries, she occupied the position of Director of Healthcare Standards within the Superintendency of Public Health in Malta, where she was responsible for the licensing of all hospitals, clinics, and residential care homes in Malta. She was a key member in the Office of the Chief Medical Officer within the Ministry for Health in Malta, leading the advisory board in charge for the introduction of new health technologies in the public healthcare system. These are just a few of the roles that Dr. Azzopardi has occupied. So the floor is yours. Thank you.
Good morning. I'm really humbled to be present here in this conference with all these experts present. You must know that I am not an expert in gynecology, and my area now is mainly statistics. So I will try to present a local flavor about the statistics of rare gynae cancers in Malta. The data that I will be presenting is data from the Malta National Cancer Registry. This Malta National Cancer Registry is a population-based registry, and so it captures all the population. We do not only collect the data from the main hospitals and from notifications by the medical doctors or through labs. We also actively seek any new cases that are incident during the particular year in study. In the sense that we also look up pathology results actively, and we try to keep our data as complete as possible. We also try to investigate the cases in order to have good diagnosis, an accurate diagnosis about the site of the tumor, and also about the histology. We also collect information about the epidemiological features and the demographic features of the person of the case. And incidentally, here we are helped because we have a unique identifier number, which is the national ID card number. The national ID card number is used throughout the whole health system, even in the private sector, and even in other sectors such as the social and social security areas. So here we can follow up cases. Our registry has good connections with the laboratory information systems of the main hospital, and we are also helped by persons who are in charge of private hospitals, clinics, and labs, because they provide us with the data. Data protection, obviously, we don't divulge data where the persons can be identified. And we collect data also on the basis that the Data Protection Act also allows us to collect data, because the new Data Protection Act, which puts into force the European General Data Protection regulations, has a subsidiary legislation, local legislation stemming from it, that allows secondary processing of data from the primary sources. The primary sources would be, for example, the doctors who encounter new cases and the labs. And then secondary processing is allowed on the basis that we are a national cancer registry that has to collect epidemiological data. And this data also is used for other purposes, including policy and planning and auditing of the health services. These are all uses which the subsidiary legislation for the secondary processing of health data allow us to do. The time period that we are going to study are incident cancers from the 1st January 2010 to the 31st December 2019. Here, we use the definition of rare cancer as those with an incidence of less than 6 per 100,000. The European Union, however, uses another definition, which is a cancer which occurs less with an incidence of less than 1 every 2,000. We also use the Rare CareNet classification for rare cancers. We extracted the rare cancers using the topography codes, the site codes, and the morphology codes, the histology codes, as specified by the Rare CareNet classification. As probably has been explained yesterday by Professor Jamagatta, the Rare CareNet classification has 3 layers. The first layer is a very broad layer, which includes families of tumors with the same referral pattern. Then the second tier is a layer which is a layer as perceived by the clinicians as a single diseases, tumor entities. And finally, the lowermost layers drills down to the particular diagnosis which correspond to the classification as defined by the World Health tumor entities in the Blue Book. Okay. Basically, this is a copy of how these tumors are defined in the Rare CareNet. Sorry that the slide might be a little bit crowded, but this is the way we extract the data from our registry. We use the Rare CareNet classification, find the particular ICD-03 topography code, which, for example, for the corpus uteri is C54, and then extract all the histological types that are mentioned. The rare cancers are mainly in tier 2 and tier 3. The table also shows the data by Rare CareNet, which shows approximately the crude incidence rate per 100,000 of each particular histological type. This is still the corpus uteri. First, we started with the epithelial, then there are the other categories. The same applies to the cervix uteri. As you might note, the crude incidence rate per 100,000 of the cervix uteri, as defined by the Rare CareNet data, it's 6.27. So practically, all cervix uteri tumors are practically rare, especially the rarest categories. This also applies to the ovarian tumors, which also have a low crude incidence rate. Here we're still seeing the classification for the ovarian tumors. The fallopian tube tumors are also very rare. Then there are the tumors of the vulva and the vagina, the trophoblastic tumors, which are very rare too, and some other rare tumors, including sarcomas of the genital and the genitourinary tract. We extracted the following data from 2010 to 2019. And between these years, we had a total of 75 cancers that can be defined as rare according to the Rare CareNet categorization. On the side, there is also the type, the histological type of the tumors that were captured, the corpus uteri tumors. The same is for the cervix uteri. For the cervix uteri, we had a total of 41 cases over these those 10 years. There is the histological diagnosis always on the side, and the numbers from each category of the diagnosis of the particular histological type. This is an analysis of the rare cancers of the ovaries. As you see, we have a considerable number of cancers of the ovaries. And these are the rare cancers of the fallopian tube. Cancers of the vulva and the vagina globally together. We had about 96 from in the 10 years under study. What I have to point out that we have small numbers. And if the incidence rate is unstable, we have to be careful about interpreting the data. We also have to be very careful in interpreting the data when the cases are less than 20, because it might be impossible to distinguish any changes from random, any significant changes from random fluctuations. And we have to be careful about interpreting and making comparisons between communities, even other countries. In other words, there is a high relative standard error when interpreting such data. This presentation also uses the methodology of combining the number of cases over 5 years so that the rates are based on a larger number of cases. Thus, we use 5-year average annual rates instead of single-year rates. Here, there is an analysis of the data between, for example, 2010 and 2014. There were 27 rare cancers of the corpus uteri in the female population. In the female population. If we add the populations at mid-year for 5 years, the total would be 1063 per the population over that period. And then we divided the rates so that we get the rate per 100,000. We have quite a high rate for ovarian tumors, as you can see. What I did is that I took a particular year when there were about 38 cases of rare ovarian cancers, and we worked out the rates per 100,000 per age group category. And also then globally for the whole population. Before, you might have noted that the crude rate was around about 15 per 100,000. And then if we corrected according to the European standard population and the World AIDS standard population, we get lower rates. That's because the population in Malta has a significant proportion of elderly persons, of elderly females, where the incidence of certain gynae tumors is higher. This is the data for the rare fallopian tube tumors and tumors of the vulva and the vagina. And there were also a few cases of sarcomas related to the urinary or to the genital system. The take-home messages are that cancers, rare cancers, are not so rare after all. In about 10 years, we saw a total of 550 rare cancers, rare gynae cancers. These cases, however, are few, considering that the cases are divided throughout the years and between the consultants. And so even the gynecological consultants sometimes would need more numbers to accumulate more expertise. So what this presentation shows is that there is scope for collaboration in actions like the GYNOCARE, where we do joint research. We establish common databases where we collect all the cases together, for example, for a European region or for the areas participating, so that we analyze them together to gain statistical power, where we can further analyze the outcomes for these rare cancers. This forum also provides a forum for the establishment of guidelines and for the sharing of expertise. A literature review recently has shown, for example, that if you analyze the outcomes, the survival for common gynae cancers and for rare gynae cancers, the rare gynae cancers always have a worst outcome. That shows that some of these rare cancers, we don't have the sufficient expertise, perhaps they are diagnosed late, perhaps the treatment options are less available within the country, because since they are rare, no policymaker would have taken them into consideration in writing policies. So this presentation shows the value of collection of data and of participation into larger forums for the benefit of patients. The main aim is to improve the outcome for all the patients suffering from these rare gynae cancers. Thank you.
So thank you for the excellent presentation. Are there any questions for Dr. Miriam? You're actually sitting next to two biostatisticians, Miriam, actually. So I'm sure that during coffee break, there will be opportunity to discuss. Okay, all right. So let's move on now. So to our next speaker, who is Professor Kleida Harasani from Tirana, Albania. So Professor Harasani, she's an associate professor at the University of Medicine in Tirana, in Albania. And basically, she's involved also in several teaching modules, such as pharmaceutical analysis and pharmacology. Professor Harasani, she is the lead for Working Group 5, which is the collaboration in clinical trials. She completed her master's degree in 2010 from Italy, and she was awarded a PhD by the University of Granada in Clinical Medicine and Public Health, and the University of Medicine in Tirana in Pharmaceutical Sciences. She forms part of the editorial board of Health Expectations, and also is the vice head for the communication research of the Western Balkans Alumni Association. Her current research interests include patient safety, oncology, and innovative teaching methods. Okay. It's all right. No worries. مش problem, no worries. Incidentally, just to remind you that those of you who haven't signed yet for today, please do sign, because as will be told by the cost officials, I mean, it's important that you sign, especially the internationals for reimbursement. Also, those of you who haven't collected their certificate of attendance, again, there are still some certificates which have not been collected yet, okay? The other thing is, I mean, for those who have joined us today, so coffee breaks and lunch will be served outside, okay? And those of you who would wish to join the tour this evening, I mean, there are already about, I think, 11 or 12 people have joined. You are still in time, I mean, but ideally tell them by the coffee break at the office opposite if you'd like to join. We're going to the old city of Medina. Last but not least, just after the coffee break, the first coffee break, if we could just gather together and have a group photo. I was meant to do it yesterday, but I forgot, but we'll do it today. So that will be before the start of the session after the coffee break. Okay, I think I told you everything. So welcome again, Professor Harasani. So who's the lead for Working Group 5, and the floor is yours.
Thank you all for coming today. Good morning. Sorry, I have to apologize. The form of the presentation is in a PDF, whereas it was supposed to be a PowerPoint, but we had a little exchange of the document. So, however, it's the same thing, but some features may be missing from the presentations. So the topic is on clinical trials on rare gynecological cancers. As you know, more than 50% of all gynecological cancers can be classified as rare tumors, defined as an annual incidence of less than 6 per 100,000 women. And such tumors represent an important challenge for clinicians. Rare cancers pose a particularly difficult issue when it comes to conducting clinical trials due to issues surrounding their clinical trial design, patient recruitment, and analysis of the study outcomes. Here is a graphic representation of the WHO classification and a full list of these types of rare cancers you can also find on the GYNOCARE website, which was compiled comprehensively by Dr. Miriam Azzopardi. In this slide, we have a graphic which was published by Spreafico in 2021, and it's a clinical trials framework from drug discovery to clinical trials and to post-market surveillance. So in phase 1 and 2, safety, tolerability, and preliminary anti-tumor activity are evaluated. After phase 2, efficacy is evaluated, typically the objective response rate as an endpoint in single-arm studies or the progression-free survival in randomized studies. As you can see, even the years below and the number of compounds from 5,000 or 10,000 in drug discovery to just one FDA-approved drug in the end. So randomized controlled trials provide the highest level of evidence to support new treatments for gynecologic cancer, and they have been key to the approval of new drugs by regulatory authorities. And a growing number of rare tumor types, each with a small number of patients, presents various challenges when it comes to testing targeted therapies, to conducting trials with novel trial design, to overcoming regulatory barriers for international collaboration, and funding for studies in rare tumors by academic bodies with little or no pharmaceutical support. These are the key roadblocks to the clinical trials, which was supposed to have an animation. But however, we can see all the key roadblocks here. And it starts with the trial startup, the lack of engagement of site investigators, of providers, of participants, the protocols that often are developed without input from key stakeholders, the protocol complexity, the high costs, especially for rare cancers, recruitment challenges, and site-based research challenges. So this all leads to fewer high-quality trials and insufficient evidence to inform clinical care. Here there are the common barriers to participation in clinical trials, and they are associated with scientists, so scientist level barriers, participant level barriers, and study level barriers. As for scientists, we can mention the lack of innovative recruitment strategies, the lack of relationships with community organizations, and a few minority researchers or staff. Participant level barriers, for example, perceived harms and fear of participants to participate in a clinical trial, mistrust, costs, transportation, the access, the convenience, the information which is unavailable in preferred language, the lack of knowledge about clinical trials. And for the study level barriers, the budgets, of course, which often are inadequate for recruitment, the lack of resources to translate, no expertise to tailor documents, and lack of culturally congruent research staff. Populations are that are underrepresented in research. That's a reality, and the FDA, for example, encourages for more participation and more diversity in clinical trials. And the non-European populations still are underrepresented in genomic testing samples, and there is a disparity of race reporting. So the objectives of our study, we thought to initiate a study, a systematic review of clinical trials on rare gynecologic cancers with particular focus on their treatment. So it's an ongoing study, which we aim to complete in a couple of months. And the rationale behind this was the Confucius saying, study the past if you would define the future. So we designed it to search the databases of clinicaltrials.gov, which is the largest clinical trials register, the World Health Organization's International Clinical Trials Registry platform, and clinical trial studies published in PubMed or Medline. So we searched for these terms related to gynecological cancers and various types, and we follow the PRISMA 2020 guidelines, which is the preferred reporting items for systematic reviews and meta-analysis. So we don't have yet the results of the systematic review. It's an ongoing study, but we have highlighted some of the clinical trials, some of the results of some clinical trials that may be included in this review. So the first one is a study of the combination immunotherapy with nivolumab and ipilimumab in patients with rare gynecological malignancies. It's a phase 2 multi-cohort and multi-site in Australia clinical trial, and the number of patients is 43. And it studies a combination immunotherapy of, as we said, nivolumab and ipilimumab. And the primary endpoint was proportion of patients with disease control at week 12 So if they have a complete response, a partial response, or stable disease. And durable responses were observed across a range of tumor types, including uterine leiomyosarcoma, uterine and ovarian clear cell carcinoma, uterine and ovarian carcinosarcoma, vaginal carcinoma, and low-grade serous ovarian carcinoma. For the entire cohort, the median progression-free was 2.8 months, and the overall survival was 15.8 months. The objective response rate of the radiologically evaluable population was 36%, and 28% was that in the intention to treat population. Overall, 31 of the 43 patients, so it's 72% of them, experienced immune-related adverse events of any grade, and 6 of them with grade 3 or higher. So as a conclusion, these clinical trials concluded that ipilimumab and nivolumab combination treatment has significant clinical activity with a favorable safety profile across a range of advanced rare gynecological malignancies and warrants further investigation in these types of tumor. This is a figure which summarizes some of the ovarian cancer subtypes and their different targeted therapies, and also the drugs that may be used in them, in their treatment. So it's a study published in 2020. These are some other clinical trials from the review. So it's a Paragon clinical trial, the ANSOG Paragon clinical trial. It's a basket trial of 7 phase 2 trials. And it studies an aromatase inhibitor, which is anastrozole, in 54 women. A subset of asymptomatic patients with estrogen receptor positive and or progesterone receptor positive platinum resistant or refractory recurrent ovarian cancer had durable clinical benefit on anastrozole with acceptable toxicity. Another clinical trial was Alienor or ENGOT, which reported the weekly paclitaxel with or without bevacizumab as treatment for relapsed sex cord stromal tumors. It included 28 centers in France, Germany, Italy, Japan, and Belgium, and 60 women participated with sex cord stromal tumors. Weekly paclitaxel is a new option for relapsed sex cord stromal tumors, but adding bevacizumab does not improve the clinical benefit. The other trial is P3-BEP trial, so it's bleomycin, etoposide, and cisplatin. It studies accelerated versus standard BIP chemotherapy for adult and pediatric patients with intermediate and poor risk metastatic germ cell tumors. It is the largest current randomized trial for germ cell tumors in the world. And the fourth one is the MEOC trial. It studies 4 different treatment regimens for advanced stage mucinous epithelial ovarian cancer. It's a phase 3 clinical trial. And the 4 treatments that are compared are paclitaxel and carboplatin, oxaliplatin and capecitabine. The third one is paclitaxel, carboplatin, and bevacizumab. And the fourth one, oxaliplatin, capecitabine, and bevacizumab. The trial stopped at 50 patients due to slow accrual and logistical challenges. So modern approaches to accurately diagnose the rare gynecological tumors are high throughput technologies such as genomics, epigenomics, transcriptomics, proteomics, and metabolomics. targets on cancer stem cells and also nanotheranostics. And this should be followed by designing of clinical trials for these rare gynecological tumors. Redesigning cancer clinical trials. Before it was one disease, and whereas today there are many different forms of cancers driven by different molecular defects and more yet to be identified. There is a new model for therapeutic development. In the old model, treatment is determined by the location of the tumor in the body without regard to the molecular characteristics of the patient or the tumor, whereas in the new model, treatment is determined by key molecular hubs that must be targeted within the cells and is only administered to patients whose tumors are found to have those hubs, potentially without regard to the tumor's location in the body. And in this slide, smaller trials have a bigger chance for success. In the old model, there were a large number of patients not selected by molecular characteristics, and there was a lower chance of demonstrating effectiveness since many participants do not have the molecular defects being targeted. Whereas in the new model, small patient populations that are all with relevant mutations or genetic defects. So there is a greater chance of desired results since all participants have the potential to respond. And here the difference between the umbrella trial and basket trials. The basket trials, we have seen one example before, test the effect of one drug on a single mutation in a variety of tumor types at the same time. These studies also have the potential to greatly increase the number of patients who are eligible to receive certain drugs relative to other trials design. The umbrella trials have many different treatment arms within one trial. People are assigned to a particular treatment arm of the trial based on their type of cancer and its specific molecular makeup. So it's like a basket with one test drug or like an umbrella with many different test drugs. So as a conclusion, the preliminary results of our review, which is not concluded, of course, show that randomized clinical trials in the field of rare gynecological tumors are rather scarce. Many rare gynecological tumors are excluded from clinical trials or studied together with other tumors from the same organ, but different at the molecular level. Cooperation between international research groups and stakeholders should be further promoted with the ultimate goal of developing better and more personalized treatments for rare gynecological tumors. Creating a reference summary for clinical trials which are finalized and ongoing will help design strategies for harmonizing new clinical trials for these types of tumors. And as a final remark, well, I invite all persons in all researchers of the fifth group, if they want to contribute to the study, we can work on this to be written more quickly or published more quickly. So everyone that is interested is invited to contribute. And also follow GYNOCARE within the social network. So we have a Facebook profile, in LinkedIn. I'm not sure in Twitter, do we have? Yeah, in Twitter too. So it's good to have a good participation and of course to also have updates of this cost action. Thank you.
So this was excellent, Cleida. I mean, I must say, Cleida is the biggest social media fan. So the minute I put something up, the minute it's her, the first is yes, and it's really important. I mean, as you know, I mean, cost action is all about dissemination and networking. And like nowadays, the way to reach people is through social media. I mean, especially if you want to get patients and other stakeholders involved. And Claudia is the number one fan. And thank you very much for promoting that. I'll be talking more about it, in fact, during the MC meeting. So any questions, first of all, for Claudia? I'm sure there will be, yes, there is, because I'm sure, you know, being public health people and statisticians, I'm sure.
Thank you so much for your presentation. It was very interesting. And of course, it's very important to reflect about the trial and rare events. I would like to stress the question of the older patients, because they usually are not included in clinical trials.
Yeah.
And it is important to take care about the elder. Also because many of rare tumors are more often present in women older than 65.
Yes.
I don't know if you consider this a problem. That is a general problem of trials.
Yes. Yes.
And
yes,
also,
sorry,
there is another funny observation. I know that the women are not keen to enter in trials generally. I don't know if it's a problem of the woman or the clinician that do not prefer to. I'm not sure about it.
It is true what you say, especially about the older people that are also a population that is underrepresented in clinical trials because they are often excluded from them due to other comorbidities or other treatments that they take and complications. So they are often excluded from clinical trials. But in order to be representative of the whole population, of course, they have to include also older people. Yes, I agree. Is the what? The family? Yes, of course, of course.
It's a cultural.
Yes, yes. Thank you.
Other questions? In fact, this is a very important point also that Dr. Gatta has raised, because I mean, it is a fact that apart from having the older population, like over 65, who are vulnerable to gynecological cancer, there's also a pediatric population who are also vulnerable to rare gynecological cancer. So by better meaning, you know, under 18 or actually before puberty. So it is a fact. And I mean, I just started working in, but I know that Dr. Brigitte Allor is involved in fact, in consortium as part of ELSI on how to recruit children and pediatrics, particularly because, I mean, I wouldn't say the majority, but a good number of gynecological cancers, of rare gynecological cancers, occur in girls, yeah, you know, under 18. And normally children are excluded from trials, of course, like over 65 were usually excluded from trials. So I know you've just walked in, no, but we're talking about clinical trials and recruiting.
There's been a lot of work by the expert committee to actually give guidelines for children doing clinical trials. So and the EPTRI is involved in this as well. So I mean, there are a lot of guidelines, and I think they do a lot of good work in Canada. Has got really their act together, where they've got all the hospitals working together.
In pediatrics.
So in pediatrics, yes.
Thank you very much.
Boston, together with Boston, I think.
Thank you. No, thank you. Sorry me, but you know, it came just in time, And thank you again, Katia, for your work on the systematic review. And yes, I mean, thank you for asking for invitations, because if at least there are 3 members from 3 different countries who contribute to this, it can be funded by cost. I mean, of course, if it's accepted, but I'm sure that it has a very good chance of getting accepted, because there's a paucity of data. And apart from that, it's one of the deliverables actually of Working Group 5. So thank you very much, Rita. Thank you. Okay, so we can move on to our next speaker. So we have Dr. Maya Sabol, yes, who is a researcher at the Division of Molecular Medicine at the Ruder Boskovic Institute in Zagreb, Croatia, where she has been working with gene-edited nucleases, using them to generate knock-in and knockout in vitro models of cancer. She is managing an ongoing grant from the Croatian Science Foundation, which is entitled Differential Regulation of the GLI Code in BRAF NRAS-driven tumors. And Dr. Sabol holds a teaching title of docent at the University of Osijek and the University of Zagreb. And she also mentors PhD students. And also, Dr. Sabol has been very active. In fact, she has published one of the collaborative papers also as part of this course. So the floor is yours, Maya. Thank you.
Hello, everybody. So thank you, Jean, for inviting me to be here. I'm very happy to be here, and it's been really nice. So I come from Zagreb. This is my institute, and it's not as grand as many of your institutes, I guess, but it's very old. It celebrated its 70th birthday last year, and the buildings are old, but now we've got EU grants to improve and to build. So hopefully in a few years, we'll get some new buildings and get things started. So the main topic of my research in general is the hedgehog glee signaling pathway. And I will briefly explain what we know about the pathway today, because it's not really that well known. It's not very a popular pathway to explore, but it's a developmental pathway that's very active during embryonic development, and then it shuts down in the adult organism. And it's inactive in adult tissues, except for a few niche cells like tissue stem cells, which keep it going and keep it active. So it's involved in this stem cell maintenance and in healing after injury, because after you get a wound, it gets reactivated and heals the wound. And it happens that it's often aberrantly activated in tumors when it shouldn't be, and it helps tumors to proliferate. And the major components of the pathway, are these 2 receptors on the membrane and the glee transcription factors, which trigger transcription of target genes. So when the ligand is not present, this receptor patch keeps the co-receptor smooth and inactive, and the transcription factors are kept in the cytoplasm in a complex that cannot move to cytoplasm, that cannot move to the nucleus, and they are degraded or cleaved into repressor forms. But when we have the ligand, which is called hedgehog, binding to the patch, then this inhibition is relieved and smoothened, can activate phosphorylation events that remove, that separate this complex and let glee go into the nucleus and trigger a variety of cell processes, all involved in proliferation and cell division. And what we know about the pathway today is that it can be activated in tumors in several different ways. So this activation can be, so therefore, described mechanisms. The first one is ligand-dependent paracrine signaling, which means that it's overexpressed in tumor cells and the signal is received by stromal cells. And this has been found for ovarian cancer. The second one is ligand-dependent autocrine signaling, which means that the cell itself is producing the ligand and receiving it. And this has also been detected in ovarian cancer. We have the ligand-dependent paracrine signaling, where the ligand is overexpressed in stromal or endothelial cells and the tumor cells receive the signal. And the final one, which is the ligand-independent autocrine signaling, which happens when there are some inactivating mutations of either patched or SUFU, or activating mutations of smoothened, which leads to amplification or translocation of GLI gene. All of these lead to constantly active signal being transmitted. And this is found in basocellular carcinoma, medulloblastoma, and the Gorlin syndrome. Gorlin syndrome is defined as it has features and combinations of both tumors and developmental malformations. But some of the tumors involved in Gorlin syndrome, beside these two, are fibromas of the ovary. So that's how this whole story started, like 15 years ago in our lab. My then boss, when I came to the labs, she said, yeah, okay, we are studying basocellular carcinomas and Gorlin syndrome patients. And now we are starting to collect some ovarian fibroma samples. So do you want to take a look at those and see what you can find? And so that's what we did. And before I go into that, I just have to stress that today we know that there's also a non-canonical pathway activation that can happen. And it can happen on two levels. So one of them is the upstream non-canonical activation, so where you have the binding of the ligand and the triggering of smoothened proteins. So smoothened can activate some other pathways beside the GLI. So it can go into MAP kinases, beta-catenin, GSK3 beta, and so on. So it can activate some other downstream targets as well. And the other option is where the GLI protein is actually activated via different pathways, and it's independent of the membrane component, so you just get GLI activation regardless of the signal. So in our studies, we have done a lot of work on ovarian cancer from various aspects of Hedgehog signaling through the years. So we started with detecting mutations in patch gene. Then we studied patch promoter methylation to see if the downregulation of this gene is associated with promoter methylation. And then we do more complex things like signal transduction, signaling crosstalk, and Indian microRNAs. And I'll start with the methylation because that's the first thing I did when I started. So it's known that methylation is one of the epigenetic mechanisms to silence genes. And if a promoter is methylated, then its expression is downregulated. So I got an assignment to screen, to find the promoter of the patch gene, to screen, to find some regions to amplify, to do bisulfite sequencing in this ovarian fibroma and also some teratoma that we had, and compare it to some other samples like basocellular carcinomas and so on. And in the end, what we found that indeed in fibroma and teratoma samples, we do have high methylation of this promoter of the patch gene, but this is not true for, let's say, basocellular carcinoma. So this is a mechanism that's, let's say, specific for the ovarian tissue, but not for skin. And to study the mutations as loss of heterozygosity, besides the regular methods, we try to implement some new things and new methods to use. And we got this high resolution melting instrument in our lab, and we were playing with it, and we decided to check if we can screen for loss of heterozygosity of the patch gene, which is also a frequent event in ovarian cancer, to see if we can see it by this HRR method, and indeed we could. So we compared the classical microsatellite analysis with the HRM, and we see a great concurrence of these two methods. And we can see that we can detect loss of heterozygosity in ovarian cancer, and the prevalence was around 30% for this ovarian cancer samples, which is lower than in literature, but this is what we got. And we also did some polymorphism studies. So we checked for a bunch of polymorphisms in the patch gene that occur. And all of these are known in public databases. And for this last one, which is the only one that leads to amino acid change, we actually found that the difference in this allele frequencies is detected in carcinoma, ovarian carcinoma, dermoids, and fibroma samples compared to healthy controls. So you see underrepresentation of the T allele and the overrepresentation of the C allele in ovarian tumors. So then we decided that this teratoma and fibroma are actually a good model to do some extra work on hedgehog signaling. And we have been trying to see if this signaling is really canonically activated in these samples. So we got some fresh samples from the hospital. We developed primary cultures, and we wanted to see if this really works or not. And there's an inhibitor called cyclopamine, which inhibits this smoothened receptor and blocks downstream signaling. And the effect is that when you have cells, I'm sorry, I don't see it very well, but GLI is present in the nucleus when you have wild-type non-treated cells. With the cyclopamine, the nucleus is empty, so it doesn't go in. And when you add extra sonic hedgehog protein as an activator, it's back in. So we showed that this hedgehog GLI signaling pathway really does act canonically in ovarian teratoma. And then we moved on to ovarian cancer. So we got some samples from the clinic of ovarian cancer, but they were not well defined of what type they were. All they said was, so we had fallopian tubes and ovarian tissue as controls, and we had some ovarian cancers of various types and borderline tumors. And what we saw that actually borderline tumors showed greater activation of this signaling pathway and of, let's say, GLI expression, SUVO expression compared to healthy controls than the carcinoma itself, because the spread in ovarian cancer was huge. And now we think it's because that when we finally got some data about these samples, it turned out they were not uniform. They were very diverse, and they were of various types, various, including rare cancers. So all this noise may be due to this heterogeneity, and we now wanted to split this and focus in more detail to a specific type, right? But otherwise, we also saw that in these ovarian cancers, we also developed some primary cultures, did some treatment with inhibitors, and what we see that the activation in these ovarian cancers can also be non-canonical, meaning that the GLI protein can be activated downstream regardless of the membrane component, and that the inhibitor that inhibits downstream components acts more potently, more powerfully than the membrane one. So that was a good, way to move forward. Yeah, this is where you can see that this lithium chloride and GANs are compounds that act downstream, and they really kill them off, right? Since we found that GLI proteins are upregulated in ovarian carcinoma, we wanted to see their downstream targets. And one of the downstream targets of GLI proteins is survivin protein, which is coded by the BRCA5 gene. And this is an inhibitor of apoptosis. And what is known is that it's mostly, it has several splicing isoforms that can be generated. And some of them are actually pro-apoptotic, some are anti-apoptotic. And we made some GLI1 knockout cell lines to check how it affects the expression of these various isoforms. And you can see, as you can see, many of the isoforms really do that get downregulated when we have the knockout, and it goes further down when we treat them with the GANT inhibitor. So these isoforms, some of these isoforms are tumor specific and can be found only in tumors and not in healthy ovarian tissue. And furthermore, they are really regulated by the hedgehog signaling. And when we did a comparison of expression of these isoforms in cell lines, which are the white circles, and in patient samples, and you can really see that these isoforms are differently expressed in various samples, but they do follow the same trend. And when we check the relative expression of these two isoforms, so this is the wild type isoform and the 2-alpha isoform in patient samples, you can see when compared to fallopian tube controls, in ovarian cancer, we get upregulation of these two isoforms. And they are, this one at least is known to be anti-apoptotic, so it promotes survival of the cells. And finally, we get to the last thing we did. And this was the one huge project that was done, where we wanted to profile a high-grade serious ovarian cancer. So that's not a rare type, but we wanted to focus as closely as we could to one cell type and exclude all the noise that we had from different rare types. And we did microRNA and mRNA profiling of these samples. So we had 16 ovarian cancers and 8 fallopian tube controls. And we did differential, we found differential expressed microRNAs and mRNAs. And we wanted to correlate this, to connect this to the hedgehog signaling to see which ones are really connected to hedgehog signaling and which ones can be associated with them. And we found a set of these microRNAs which were of interest to us. So these are all either components or downstream targets of hedgehog signaling. So the hypothesis was that the hedgehog signaling seems to be downregulated in high-grade serous ovarian cancer compared to borderline where it was up. And this downregulation could be caused by overexpression of these microRNAs. And if you manipulate with this microRNA expression, can we affect this biology of these cells? Can we change the expression of targets? And first of all, we validated by qPCR the findings we had on microarrays on extra additional samples. And we found that we actually excluded these two. They were not relevant in this setting, but we had a few of them that really were upregulated. So the microRNAs are upregulated and the gene is downregulated. So in the same subset. And we did some experiments on cell culture to see if this really goes the way it goes. So if this microRNA really does control the expression of these genes. The first thing we did was the luciferase reporter assay on 2 ovarian cancer cell lines to treat them with mimics. And if the mimics work, then the expression should go down, and it does for these. And we did gene expression analysis, so treated the cells with either mimics or inhibitors. And as it says here, we got what we were expecting. So we validated, let's say, 3 microRNAs and their corresponding targets. And we also are doing, and we're doing cell viability assays, migration and colony formation, which I will not show here because it's a lot of images, but it all fits within this story. And the final thing that we did to prove the biological effect of this thing is to use the chick chorioallantoic membrane assay, or CAM assay. So this assay is a proxy or a substitute for in vivo experiments, because what you do, you take cells and you plate them onto open eggs, on fertilized chick embryos. You let them incubate for a few days, and then you harvest the tumors and harvest the chick organs and see if you get invasion of human cells into chicken tissues, and if you get a tumor invasion through the CAM membrane. And this is what the CAM membrane looks like. So this is the membrane itself, and these dark purple blotches are tumor cells. And this big thing is matrigel that we plate on it. And the cells from matrigel do go into the CAM, and they do invade inwards. And when we do, let's say, statistics of this, unfortunately, even though there is a trend that we see, this is not significant statistically, but maybe if we did more samples, it would pop up more. But anyway, compared to control, the inhibitor reduces tumor size, the mimic increases tumor size, right? And right now we have collected brain, liver, and heart from these chick embryos. And we are doing quantitative multiplex PCR to detect the presence of human DNA within the chicken, to see if the tumor cells invaded into the chicken tissues. And we expect invasion into the brain and liver, because ovarian cancer tends to go there, but not in the heart because it doesn't go there, right? And this is being done right now. We had some issues with this assay because, first of all, it was done right in the middle of the epidemic, the pandemic, so we couldn't go to the Faculty of Veterinary Sciences to do it. So it was when the restrictions were off, we went there. We injected a few eggs and then we ran back, and then for a few weeks or months, nothing, then again. And this has been really labor intensive. And finally, so at one point also, I don't know if you're aware, but just as the lockdown started, we had a massive earthquake in Zagreb, and a bunch of these samples was lost because they were in pathology, they were ready to be, put into paraffin blocks and so on, and everything fell, everything crashed, and most of it was lost. So now we have to rely on this bit to show what we wanted to show. Yeah, and as for what we think it's relevant here for cost and in the context of our work, so what we figured out during all these years is that sample characterization in general in papers, it's not good. Not hours, we can start with hours and then go on. So in many papers, still you only get OC, like ovarian cancer, you don't get any subtypes, you don't get any classifications of rare cancers. And this generates noise. And we cannot, sometimes we cannot get clear enough results because we get this too high variability, too much noise, and maybe because of the inclusion of these rare types into the set. The third thing we are struggling with is a small number of samples per study, because in Croatia we get maybe 400 ovarian cancers per year, and that's throughout the whole country. So our collaborators can provide maybe 50 if they are very, very eager, and often they are not. So we have to rely on clinicians to get us samples, and it doesn't work very, very smoothly as it should. So we get studies with small number of samples, which are not very convincing, and then we have to make do with other methods. Also, sample type and quality are sometimes the issue, because okay, we get fresh samples. Sometimes they are not preserved well. Sometimes they are not immersed in the solution that they should be, and then RNA quality is bad, and then we cannot do stuff. So often we rely only on paraffin embedded tissues, or if we're lucky, then we get some nice fresh ones to work with. And as we are not a hospital, again, it's difficult to obtain samples as is. And the paperwork for obtaining patient data is also hell. So we have to go through several months of different ethics committees and whatnot just to get access to the clinical data we need. And of course, funding, I guess, as is with most of us, it's irregular and it's limited, and hopefully inclusion in this cost can help us in that regard, right? And just to acknowledge all the people that have been involved in this. So this is our lab. So Diana is a postdoc that recently left, and Sonia was actually the PI, the boss of all of this until so she retired 2 years ago, and now Petar took over the lab. And we are still, we are growing. We are trying to get new people to get interest in our work, and we have running collaboration with some of the clinical institutions and some hospital centers in Zagreb, including the Institute of Pathology. And I'm sorry to say that this doctor, she passed away recently, so now we have to find a new pathologist to help us with all this, because she was one of the few people that could analyze chem assays. And so this chem assay was obviously cursed. And we also had some international collaborations. So bioinformatics division at Innsbruck they were helping us with analysis of this microRNA data. And we had some help from Professor Seilinger for samples. And this Professor Nielsen did the follow-up of our methylation assays, and we sent him some samples to do methylation on his own. And these are the funding bodies. We are proud to say that we got some Terry Fox, grants, so they recognized us as something relevant, and the Croatian Science Foundation. And I just wanted to invite you, if you're interested, we have a meeting in Zagreb in November of the Croatian Association for Cancer Research. And we usually have speakers, international speakers, including the EACR members. So it's usually half Croatian and half international speakers. So you might be interested, and you're all invited if you would like to come. Thank you.
So thank you very much, Dr. Maya. Very interesting. And certainly we'll keep in mind that date of November, so we see whether we can do a combined working group meeting or workshop or something, because that's very good. Any questions? Yes, I expected there will be questions. So
I would like to know if you are connected also with the European Reference Network, EURACAN, where there is a domain of rare gynecological cancer. The leader is the Isabel Rey-Cocar. And in this group are also made this type of studies. So which are very important to understand the reason of several, the problem of aggressiveness of some diseases with respect to others. And this is important also because a very collaborative group, the European Reference Network are the European network that for me is very important to be connected with together in order also to understand which kind of trial or study it's important to be conducted, and not only the usual for the richest countries, which is not the case of.
Yeah, thank you. I did not know about this, but I will ask you for the contacts later, so I can write it down.
As a suggestion also for this group, which is a very
great. Thank you very much.
Thank you. I enjoyed our talk. It's not my, but I was interested in the borderline tumors because for us pathologists, we really look at them as a risk, but they were behaving more like the benign. So I was wondering if that's true or not. I mean, your numbers were small, but maybe what you're doing could actually provide some sort of test to see when these borderline tumors actually become malignant.
Maybe, yeah. So this was a small study because we had a small sample number, and we would like to expand this to a larger set and see
and
if it holds true.
There are many different types of borderline tumors.
Yeah. But it was surprising for us because we were also expecting for the borderline to be
malignant, behave like the malignant ones.
No, to behave. So in our study, it turned out that they were behaving more like malignant and ovarian cancer, right? So they had higher expression of Hedgehog signaling, and they were more upregulated, right? So, and our clinicians were trying to convince us that no, they should be somewhere in between the ovarian cancer and
because histologically the early ones look very similar to benign. So it's interesting to see when actually do they become malignant or not.
Yeah.
That would be the crucial thing. Thank you. But it's a lot of work to do.
Yeah.
Hi. I wanted to ask you about the different isoforms that you. Because you mentioned one target, right? And you should look at the relative expression of one target. I was wondering whether all those isoforms are expressed in the nucleus, or did you look at whether they are actually sequestered at the cytoplasm, since the
we didn't look at the localization because we had troubles with the antibodies. So the commercial antibody, it recognized some of them, but not all of them. So, and when you put the antibody on the cells, you cannot really know what you're catching, right?
Okay, and in terms of nuclear localization signal, is it always in all those isoforms?
It has it always. So let me go back. I didn't mark it here, but it should be in the beginning.
It's in the beginning.
Yeah.
Okay, no, fine.
Yeah. So not much is known about this splicing isoforms. This was one paper that my colleague Vesna ran into, and it said that different isoforms have different expression in tumors, and some of them are present only in tumors. And now we are also doing in parallel some studies on breast cancer and on head and neck cancer, and it's the same thing. So some of the isoforms are really present only in tumor tissue.
Because it might be worth doing, since some are pro-apoptotic and some are anti, doing a ChIP-seq of the different, if you have a good antibody or which you don't. So maybe tag them and then do a chromatin immunoprecipitation to see the different targets, because that could really reveal whether there is a difference in transcriptional activity.
Yeah, good. Thank you. Good idea.
So this is in Dutch. It shows here the presentation.
Okay.
I didn't know, and I have taken all the written things, but you can show it.
So it's a touch option.
The presentation was shown here. It was very comfortable.
So actually, you just
there and
Yes.
oh you did something.
And you can
No, I don't think he controls it.
but you can change from here.
Oh, and also you have the remote.
Interesting.
Very nice.
But it was a nice work. Your presentation.
Eu lovely como share na všechny application centers.
Mela, mbagħad inti jiena kif m'għandix, ġifieri inti mbagħad x'ismu x'in mbagħad nippropon naqra PDF tal-presentation. U neħi li ma dak
Jekk jogħġbok, Iva. Ikkupja, imma bad tixxerja jekk jogħġbok għax ħan emenda qabel tapplodi għawa. Kemm inneħħi żewġ slides.
Mhux se nixxerja tibżax u Jonathan kemm
Xtaqt niġilbirek ukoll għax taqalu ta' Prof Felice. U xtaqtu jkun għawilu, ma Le, mux ħana emenda s-sana, qabel t-xerja.
Jena tibza xina mbaċi ħan nitlobom il-PowerPoint ħajtuni minn ħajnħiġi data, ħajnħiġi data, ġifir tinkweta
Inti ma t-xerja fix marru.
Le, ma mux se jibbatom, eżat, Jonathan, minn ħat l-ofistani għall-IT, xerifet juħusib Iwa, mela, yes.
U jek tamla presenter Nikolaj tajjuna. Le, għawnek biex tidher bil-notes. Ma jimportax, ħan ġib It is a good virtually. Okay.
Sharif għandu man flowly di David Saliba and clear we had għandu Sharif tal-BD but then we had sorry then we had Manġunata David l-ektar wieħed reċenti. Third more efficient. Mandu problem. So the newest with the most problems. Lara x'jiġi ma jitxjaq bidel il-part għan jessel għandu warranty ma jkollhom jaħdu balla flus. If it's under warranty, għax ta' spejjeż tagħhom. And no, they try to store as a company. Have this second double warranty? It's more like you know, if you don't emphasize or push, nobody will do anything.
Okay, so thank you very much. I hope you enjoyed your lunch break with the very healthy, fat-burning pastizzi. But they are the Maltese delicacy, you know, and like it's part of the reason why we are a bit obese, most of us, but they're good. And also, just a reminder, those of you who joined us later in the morning to remember to sign, okay, for today. And also, if you haven't collected your certificate, to get the certificate as well, okay? Okay, so it was also very nice seeing different groups networking with each other. That's the whole scope of cost, all right? And something which you can only achieve when you meet physically. I mean, not so much. I mean, you can try to do it virtually, but it's not the same. So, and I think we can move on now to the next speaker for this session. And I'm very happy to introduce a colleague of mine, work in the same department, and this is Dr. Nikolai Pach. So Nikolai is a senior lecturer within the Department of Anatomy, and he's an investigator in human genetics at the Center for Molecular Medicine and Biobanking in the Faculty of Medicine and Surgery at the University of Malta. Dr. Pach obtained his PhD from University of Malta, has been working on genomics of risk stratification, particularly in type 2 diabetes, but he's involved in many, many other projects. His current research interests focus on medical genomics, molecular endocrinology, the application of omics to rare disease medicine. He's currently leading several research projects on genomics, which basically span from inflammatory bowel disease, diabetes, adenomas, and others. And very importantly, has recently been appointed as the National Node Director of BBMRI, and is a working group member of the Million Genomes Project of Europe. He's been awarded several research grants, including the University of Malta Research Excellence Grant Award, and he holds active collaboration with many clinical partners, both locally and internationally. So the floor is yours, Nikolai. Thank you.
Okay, thank you. Thank you, Jean, for the kind invitation and the introduction. So I'll be discussing population-based biobanking in the context of genomics of the Maltese Islands. And as Jean said, I'm recently quite actively involved in the relaunch and revamp of the existing biobank structure within the University of Malta, with the aim of reaching a very specific goal that I will outline towards the end of the talk. And I plan to sort of skim and over merge four related concepts. So first of all, why do we focus so much? Why is so much energy devoted into population-based biobanking, especially in the so-called post-genomic era, we still don't know that much about drivers and progression of common complex traits. And obviously, how does this link to the genomic architecture of a population which is not so strongly featured in genomic studies, the genomes of Malta, and our plans for Duana Bio, which I will comment on towards the end. So starting off with some very basic points, but what should a biobank deliver? So we can all agree a biobank means different things to different people. And you can call many sorts of collections or repositories a biobank, but a biobank should be able to deliver some core outcomes. It is obviously a repository not only of biological material, tissues, fluids, or blood or DNA, but also of clinical epidemiological data, electronic health records, and other information. Biobanks are dynamic, so they are not static, fixed in time, but they are long-lasting, long-running projects, and they should be tied to a specific objective. It makes very little sense or no meaning to collect samples without a specific objective in mind. And obviously, these 3 points apply to any collections of samples and specimens in a laboratory fridge or freezer. But what distinguishes a collection from a biobank is that biobanks should have the data management infrastructure, and there should be the governance structure to facilitate sharing of materials with outsiders outside of the biobank. There should be appropriate quality control procedures and obviously oversight from ethics review committees and obviously other governance structures that are in some countries mandated by law. So what's the relevance of a biobank in the modern day era? And if you go back a bit in time, biobanks were not so crucial, but what spurred the development of large-scale biobank studies, such as UK Biobank, the Estonian Biobank project, and many others, was a technological advancement, the ability to investigate and interrogate thousands of polymorphisms in large numbers of cases and controls. So suddenly people realize that you cannot really look at 100 polymorphisms in 200 cases and 200 controls, but suddenly it became possible to look at 500,000 polymorphisms, for example, and to reach statistical significance there, to correct for false discovery, to correct for false positives, you need very large sample sizes and obviously a correspondingly low p-value. And this spurred the development of these large-scale infrastructures, and this raised obviously a number of ethical, legal, and social issues, which I gather were discussed yesterday. But very importantly, biobanks came to the forefront, and the social dimension of a biobank became more prominent, whereas a biorepository was previously very much uncontroversial, residing within the repositories of a pathology department in a hospital. Now a biobank became very visible, and it raised a number of societal issues as well as data protection and legal issues that are still very relevant. But if we put all those aside and focus on the science, what should a population bank do? Basically, a population biobank at a very simple basic level brings together, it marries, it merges a collection of multi-omic data, because that is the direction that most research is taking nowadays, with clinical and lifestyle factors. And a population-based biobank should, in my opinion at least, be able to deliver 2 basic outcomes. It should facilitate the research which aims to uncover disease biomarkers. And for this, you require prospective epidemiological long-term follow-up studies. This is the brilliant job that UK Biobank is doing. And obviously to do so, you need to recruit healthy donors from a representative region or country. And secondly, a population-based biobank should, at the very least, least, be able to deliver insight into allele frequencies within a specific population. And the best paradigm one can make of this is perhaps in pharmacogenomics, right? So there is a large number of genes which heavily determine patient outcome, and the clinical utility and economic utility of testing for these genes in a population depends on how frequent they are in a specific population. And we know that you cannot generalize findings from for example, North American populations to, for example, a North African population. It doesn't work that way. That's a point that will come up shortly. And tying in with my earlier point, if you visualize disease development along a trajectory, so right from the time of birth onward, so we are all born with a genetic susceptibility. There is an environmental exposure which acts in a dose-dependent manner, and then somehow, somewhere, no one knows why, a threshold is reached. And you get an early lesion, subclinical disease going on to clinical disease, and then precision medicine comes in here, diagnosis, prognosis, and response to intervention. And there are obviously very good clinical-based biobanks focusing on specific collections, but if you just focus on disease biomarkers, you are unable to address these 2 points here, the biomarkers of exposure and biomarkers of susceptibility, genetic susceptibility or otherwise. So a modern research strategy actually draws on these 3 basic points, and to do so, you need a population-based biobank. So again, this is not my words taken from a publication, but you can imagine the concept. So numerous disease-specific biobanks focusing on specific pathologies, and in theory, a large population-specific repository that is disease agnostic. So it's a reference population that is matched to the population being studied. So we integrate that with multi-omic data sets, health records, electronic health records in particular, and other phenotypic information, and that can provide new insight into treatment response and drug targets. And if you focus a bit on the difference between a clinical biobank and a population-based biobank, then obviously there are excellent repositories of residual tissues or disease-specific biobanks which are in hospitals or academic centers, but those cannot be used to provide true estimates of allele frequency in a population. And again, biobanks composed of single gene disorders, again, similarly cannot be used to estimate allele frequency. So there is this very significant need for a population-based biobank. But you might wonder, so what? Isn't the UK Biobank around, isn't repositories such as GNOMAD and AGSEC and TopMed and all the big projects have been done, aren't those generalizable to other populations? And the answer is quite simply no. And I will discuss why fairly shortly. For those of us outside of the molecular genetics field, at GWAS, which the GWAS revolution, which spurred the development of a biobank, basically involves genotyping tag polymorphisms, thousands of them from across the genome in large numbers of cases of controls. Then obviously you correct for false discovery, by lowering the p-value, you come up with your fancy Manhattan plot, you explore regional linkage disequilibrium, and then obviously you need to replicate that association in an independent collection. So there are here, this is showing us why we need thousands of samples in order to replicate and discover genetic associations. And obviously, and that's not even going into the need to biologically validate associations to mechanisms. And what most GWASs have told us is that for many complex traits, you get large numbers of common variants with very tiny effects, odds ratios of 1.1, 1.2. And that makes clinical translation fairly a bit of a headache. And what have GWASs delivered? Well, they have been very successful in Northern European populations, where most GWASs have been conducted. They have focused on easy to measure phenotypes, so BMI, waist circumference, diabetes, but not so much the difficult phenotypes. Think of neuropsychiatric disease, for example. And they have focused on simple genetic models that are easy to compute or relatively easy. But then there is the whole area of the iceberg beneath the sea level that is still relatively unaddressed. And again, not all of these questions can obviously be answered by population-based biobanks, but you can see that one of them or two of them focus on the need for larger, more ethnically diverse reference panels. And I want to emphasize that this is not simply a question of academic relevance, because whenever has direct clinical translation, because populations which are underrepresented in large data sets such as GNOMAD will have and do have a different genetic architecture. And whenever you try to score a variant for pathogenicity, populations which are underrepresented in GNOMAD or AGZAC have a much higher risk of returning variants of uncertain clinical significance than a simple, straightforward classification. Is it benign or is it pathogenic? So there is direct clinical relevance to the rationale for driving the need for large ethnically diverse reference panels. And some people have argued, but has this population scale big data delivered? And if it has not, then why should we keep pursuing this track? And there are two simple examples, not to make a big fuss about them, but for example, hypertension and autistic spectrum disorder, they are two very heritable conditions. Billions of research funding has gone down this path. And as yet, over the past, 10, 20 years, no new drugs for hypertension, and we still don't stratify treatments according to specific genetic architecture. And you can say the same thing about the complex spectrum of autistic spectrum disorder and related traits. And what is being missed? There has long been this big emerging notion in genetics that we are failing in our diversity. And you can see some points here. It's perhaps a bit too much of a busy slide, but individuals of African and Asian ancestry are definitely more likely to receive results which are of uncertain significance or unambiguous genetic results. So after all that money that goes into and energy that goes into obtaining a full exome, for example, you come up with an uncertain significance. How do you explain that to your patient? And that is very, very challenging. And to make things worse, associations which have been identified in European populations might show conflicting or opposite directions of effects in others. So clearly, the worldwide heterogeneity is widely misrepresented. And the interesting thing is that this was initially rationalized on the basis of recruiting an ancestrally homogeneous population. And this, I'll skip this, but this perhaps promotes a simplified view of a European continent population being fully homogeneous, whereas we know that it is not so, and there is complete or nearly complete lack of understanding of global population structure that prevents correction for stratification bias, and obviously an unequal representation of diversity within major genetic data sets. And again, this is what I was referring to. This is GNOMAD, which is what I believe it's used in automatic fashion by most variant classification algorithms. You can see that the overwhelming majority of individuals there are Northern Europeans. And I came across a recent paper up to 2019. Most GWAS involved, I believe it was close to 80% either North Americans, British people, or Icelandic individuals. So a very big failure of genetic diversity. And against all of this background, how can population banks contribute? So obviously, you know where I'm going. There's an increased demand for samples from a more diverse range of population, but also some other technical issues that are perhaps a bit more boring. Diversity, quantifying diversity, comparing it and annotating it across biobanks is also very challenging, even mathematically. For example, the accuracy of PCA has been challenged, and this is not easily acknowledged. How can biobanks share and compare summary ancestry statistics from diverse data? And obviously, like all other things in science, the tools that assess genetic structure offer various strengths and limitations. So against all of this, what do we know about the origins of the contemporary Maltese? So there was a brilliant paper published a few days ago showing us that temple people of Malta were inbred and short. We now, well, we're still short. I won't comment on the inbred, and now we're obviously fat. But where is this coming from? So contemporary Maltese are without any doubt Southern Italian. So contemporary Maltese, according to one of the few studies actually by Capelli, they originated from settlers in Southern Italy. And obviously we have a very colored history at that I'm sure you're aware of. So multiple conquests, depopulation, immigration, and repopulation have obviously affected our genetic legacy. And we are a fairly outbred population now. But and I know this is contested by some people, there is also Middle Eastern contribution, specifically from the Levant. And this is largely the fault of the Phoenicians, the Phoenician settlers and traders. No one knows, people don't know that much about the Phoenicians, but they traded quite actively in the Mediterranean, and they established trading bases in Carthage and Malta, and they do have an effect on the legacy of the Maltese, as is also quite relevant with the case of the Italians. So there is this shared overlap. So this is showing you correlation between the Toscani in Italy, the 1,000 genomes TSI dataset subset, and minor allele frequency in the Maltese for, I believe this is 200 or close to 200 pharmacogenetic SNPs. And based on all this, what do we know about the genomes of Malta that really warrants a better understanding and the need to push for a population Maltese reference collection? And this is unpublished data, so please don't run off with it, but the Maltese population demonstrates this shared overlap between European and Middle Eastern genomes. And you can see this is PCA, mapping on PCA components of data from around 550 low throughput whole genomes from diverse collections in projects that I currently lead. And you can see the black circle, that's us, the Maltese, and shared affinity with the Europeans in the north and the Middle East component in yellow. And this is on data from HGDP, the Human Genetic Diversity Project. But then if you explore the fine scale structure, this is now the genetic data is imputed, it is plotted on PopRes data from Europe, and you can see again the Maltese in black. So quite some of the data points scatter over the southern and southeastern Europeans, but fairly distinct from northern and central Europeans. So one take-home message there is that there's this shared overlap between European and Middle Eastern genomes. So we are partly unique. Research also demonstrates that there is a significant difference compared to the rest of Europe in risk allele frequency for complex traits. And I'll take one example about NOT2, inflammatory bowel disease, one of the top most studied complex phenotypes. I believe over 300 loci linked to IBD, probably more now. And NOT2 was the first. And NOT2, if you look at this paper actually presents very strong odds ratio in a dose-dependent fashion for NOT2 variants, for loss of function variants in NOT2, odds ratios of 17. So that is very, very strong. And NOT2 is expressed in the Paneth cells of the small intestine. It regulates this intestinal microbiome, and it's very unique because it strongly increases the risk of ileal Crohn's disease involving the ileum, but not of ulcerative colitis. And if you look at some of our data, we picked the 3 polymorphisms, loss of function polymorphisms that are I believe it's between 50 to 80% in Northern Europeans in ileal Crohn's, and they are simply not detected in cases of ileal Crohn's in our data set. So this is a data set of around 500 patients. So quite a significant difference. And obviously, we didn't just stop there. We carried out our own mini GWAS, and this also involved the same cases and controls on inflammatory bowel disease. And you can see in Manhattan plot some very strong signals. And you can see in the quantile plots no evidence or little evidence of forced stratification, but some very strong signals. And if you investigate these further, so we actually detected in a relatively small subset of patients, but these are, let me put this in perspective, these are nearly all of the patients with Crohn's disease and ulcerative colitis on the island. So it is quite well representative of the disease spectrum here. And we detected some signals that were published by other Northern Europeans, some signals that were not there, but have been later picked up by the first trans-ethnic GWAS study, thereby highlighting the need for multi-ethnic studies in most complex traits. And very interestingly, some signals which have been associated with autoimmune pathologies related to inflammatory bowel disease in other smaller studies. So there is a different genetic spectrum when it comes to risk alleles for complex traits. And the third point is also quite relevant. This is the work of my colleague Ruben Kauki on amyotrophic lateral sclerosis. And ALS is also on a spectrum, just like diabetes, so monogenic versus polygenic, familial versus sporadic. And Ruben quite elegantly showed, and his researchers showed, that in the top 4 genes associated with familial or sporadic ALS in Northern Europeans, then again, none of the Maltese patients carried deleterious damaging variants in these cohorts. So what does that imply? Is this red component here, the unknown, is it larger? So probably it is, but basically the genetic cause of 2/3 of familial ALS cases cannot be pinpointed to known ALS loci. And there are other examples of this. This is some of work of my students and my colleagues. We have a very strong interest in rare subtypes of diabetes, so phenotypic extremes typified by monogenic diabetes, but also the monogenic metabolic syndromes, which are quite rare. So last year we picked up close to 150 samples, individuals with early onset impaired fasting hyperglycemia in the non-obese state. And that's not an easy task in an obese population. And we asked the question, how many of them have a monogenic variant? And quite simply, you can see 4 or 5 variants came up. Most of them, bar one of them, bar E256K, were novel. So novel variants, so much so that we had to go into the trouble of actually performing enzymatic assays to investigate the catalytic activity. And surely enough, to our delight, these variants were all associated with reduced activity of, in this case, glucokinase. And we also ran into problems here, because even clinical criteria that are used widely to identify subtypes of disease, common traits for genetic studies, these were developed in the British, right? And extending and demonstrating their utility and validity, sensitivity and specificity in a Southern European population where there is variation in genetics, but also variation in, for example, background prevalence of obesity, for example, that has to be done. So clinical scoring algorithms have to be refined. So also people go about saying nowadays that we need to identify phenotypic extremes, but what defines a phenotypic extremes? And there are other examples. My colleague, Professor Alex Felice, was here yesterday, and he, I'm sure, as you well know, is a hemoglobin researcher. And we do show evidence, the local population, for enrichment of deleterious alleles. So I will not discuss thalassemia, but again, Professor Felice and Chris Sherry and their teams have over the years shown that most of the cases of thalassemia locally are attributed to a handful of polymorphisms, but that is also relevant to the Mediterranean. But what about other complex traits? And again, one of the projects that we are currently involved in is hidradenitis. And hidradenitis, this is a chronic inflammatory disease of the pilosebaceous unit, and this is quite common in Malta. Prevalence rates are much higher compared to the rest of Europe. And you might wonder, what's genetics got to do with this trait? Well, actually, it has to do quite a bit, because mutations or variants in gamma secretase. And last year we stumbled, quite by accident, I must say, on a lean patient, non-smoker with atypical HS. And obviously, we selected this for further studies, and we came up we identified an in-frame deletion of 4 amino acids in this patient in nicostrin, which is the unit in red, and this interacts with PSEN2, which is in yellow. And that was, this is monogenic HS. And to our surprise, obviously, we didn't sit on this, but we looked further, and we have to date nearly sequenced 100 patients with HS. And to our surprise, this is also unpublished data. The same deletion shows enrichment in cases with atypical forms of HS. So typically very lean with atypical features of the disease, and it segregates across generations. So you can see here the patient's mother with also more or less the same pathology. And finally, we do show evidence for enrichment of the literary alleles. I will not emphasize this a lot, but this is Netherton syndrome, for example. And we stumbled on this also because of a very sharp eyed colleague, clinical colleague. Netherton syndrome resembles eczema in kids. It's autosomal recessive, and you can see this is normal, this is abnormal tissue from skin. And we, a few years ago, detected a novel splice donor variant. It's away from the canonical donor site, the GT here, so it's around 5 bases away, so it's still relevant. But you would not believe, or we could not believe, when both, where obviously the child was homozygous, but both parents were heterozygote carriers for a mutation for this variant that was novel, not described in GNOMAD, and the two are unrelated. And we had quite a hard time convincing the reviewers of this, but two unrelated individuals, as far as they know, carrying a rare, a novel intronic variant that was not even present in our ethnically matched reference data set. So again, this provides some compelling evidence for the need to establish a reference population genetic data bank. And quite coming to an end, I will propose now what is being discussed. Dwarna Bio. Dwarna is in Maltese. It means about us. So in typical Maltese fashion, it is shamelessly stolen from all of us in the United States. Dwarna Bio is a national initiative being led and proposed by many of my colleagues at the University of Malta to establish a central population-based biobank. And I've added the word genomics there because a bank has to start from somewhere. So although we plan on expanding, we need to focus on one achievable goal. It is obviously a major logistical undertaking to establish a nationally representative collection with consent for multi-omic analysis and biomarker and epidemiological data. And we hope to establish a prospective cohort dynamic design. And I'll point you to a paper that my colleague JP Abeyer and Bridget and many others wrote on the European Journal on Dwarna. Dwarna is the portal. It is this blockchain-driven mechanism that gives participants access to information to control how and where their samples should be used, and obviously presents a solution to the GDPR right to erasure. So the vision is to leverage the power of a tiny island where we have one major hospital, one university doing research. So that is within itself quite powerful. So to build on that, to overcome issues with regards to quality management, pre-analytical effects, custodianship of existing clinical collections, and we hopefully think that this should act as a seed for multi-omics longitudinal cohort in a study with different mechanisms. We are part of the BMRI ERIC infrastructure, so ideally the data management and governance structure overlaps and is shared with that. And we do envisage governance and cross-sector collaboration with other resources that are untapped. So a histopathology tissue bank, for example, that should be integrated into or we wish to integrate, as well as an imaging bank and the upcoming 1 million genomes of Europe, to establish a medical genome reference bank. This is something which I will keep pushing for, because this directly impacts the deliverance of genetic service in the island, and obviously an independent access committee to regulate access to the bank. There are obviously challenges that we are aware of, and I will leave you with this. There has to be funding to ensure long-term sustainability. We currently have only short-term funding, and many biobanks run into trouble with commercialization or industry partners that we wish to resolve or avoid. There has been no, unfortunately, feasibility study, and this worries us quite a bit, at least it worries me, in anticipation of the process of recruiting volunteers. We don't know whether this will be a success or not. The acceptance of the research vision and trust in the safeguards for participants remain a key issue, and these should be maintained at all costs for project success, and it has to build on a vision for promoting health for society. We do not envisage the country going anywhere enacting a biobank law. These are things that take quite a lot of time, unfortunately, so unlike what the UK did. There are also LC issues, but those are outside my expertise. And there is sometimes this feeling that there is less lack of appreciation from the general public on the research utility of a national-based population biobank. So that's all I have in mind. I hope the next time we might meet, we will comment on the success of Dwarna Bio. Thank you. Thank you.
And I'm sure there are some questions. So, so you don't think, no? Yeah. Yes. So.
I understood from your presentation, very, very good and rich, that the meaning of population-based biobank, population-based, is different from population-based cancer registries.
Well,
because
yes.
No, it is another, I reflect about this. You mean biobank coming from different part of the world. So in order to understand the differences among different parts of the world, but not the exhaustive, the representativeness of a specific population. I mean, for instance, to define a sample of the specific population you would like to study, and invited the people to give some...
Exactly. So, okay, I understand that biobank means different things to different people, but what we had in mind, at least what I had in mind, is the UK biobank model. So you identify a segment of the population, you invite them to, they are screened by healthcare professionals, they're invited to donate a sample, and obviously following the correct ethical procedures to donate a sample for research. And then the participant can select, say, I want to participate in any research, or research on heart disease, or research on, I don't know, osteoporosis, or a specific pathology, or they can ask to be recontacted if a new project comes along. But a population-based biobank aims to create a representative collection of the population out there, so it shouldn't be targeted to one specific disease. This is the UK biobank model. I keep keep referring to it because I admire a lot what UK Biobank has done, and some very big papers coming out from UK Biobank. Now it's been years down the line. So, and I hope in a few years' time it will be our legacy to the people of Malta and to the university to establish this. It is academically relevant. It is relevant at the public health level, I guess, as well. So I hope we'll get there, but it's not a disease-specific bank. No. But it doesn't mean that it should not collaborate with specific disease biobanks. But we distinguish between a population bank and a clinical bank. So there's no reason why the two shouldn't be interoperable. They should not share data and participants, but they are different. They answer different research questions.
Okay.
David.
Hi. Great talk. Absolutely. Thank you. I just wanted to ask you about, since you mentioned Crohn's, one thing that comes across in Crohn's, but many autoimmune diseases and celiac is the HLA class 1. And you see it like the most, everybody they do these huge GWAS studies and then HLA class 1, which is obviously the most polymorphic. But I wonder whether you have any insight.
Yes, in fact, you look at the Manhattan plot, there was this big peak around chromosome 6, which is where the HLA locus is. So I believe you have to count it because 1, 2, 3, 4, 5, 6, so it's the red. And there were a couple of HLA loci. This was done on immunochip. So immunochip doesn't provide exhaustive genome-wide coverage, but it is specific for autoimmune pathologies. This was not sequencing, this was immunochip data, but there were a couple of HLA loci. You'll have to excuse me, I just picked this graph out of a paper and a dissertation.
In something terms of the Maltese population, what's because obviously again it's the most polymorphic, how does it, is there any data on that?
On HLA loci in the Maltese, it's not presented here, but we can discuss that because yes, certainly. We've got a wealth of genomic data that I keep saying this to many people, we're sitting on a lot of data, genomic data that could serve different purposes. But I recall, I will forward you this dissertation if you'd like on the paper. There was a big, there was definitely a big signal coming from chromosome 6 when HLA is located. So I hope we discuss that later.
In fact, I would, first of all, thank you very much. It was excellent. And I would like to stress the point that being a small country, and I mean, Malta is small, but other countries like Montenegro, Moldova, I mean, so many other small countries, where by being small, especially when it comes to creating biobanks, they have their advantage by being small because you can have a whole nation.
If we can't manage to do it with one hospital, one university, then it really says a lot about
communication. Communication, exactly. So, and again, I think, in my opinion, that small countries, especially, or countries who are otherwise maybe, because they're small, there might be a bit disadvantaged. By being part of a network like this, that's where you can learn from each other. Like, for example, the patient recruitment. And we had a talk yesterday by Dr. Sharon O'Toole of her experience in Ireland, which she based it from experience in the UK with the GYNOCARE Biobank. And learning from her, I mean, got a lot of people interested in knowing how to reach out to patients and make the patient like a partner in biobanking.
And this is something that Dwarna Bio has to address very soon, in fact. And it involves people coming from outside the field of research, I must say, because we researchers, at least I as a researcher, tend to see things as from one perspective, whereas that might be very different from, we've been told that we see patients as walking samples sometimes, and that is, that's incorrect. So, and I know that's incorrect. Thank you.
So our next speaker is Neil. So Dr. Neil Conlon from Ireland. So Neil is a research fellow based at the National Institute for Cellular Biotechnology in Dublin City University in Ireland. And his research is centered on repurposing approved therapies for the treatment of hard-to-treat cancers and focuses on hair family-driven cancers, including ovarian, endometrial, and breast cancer. He received his BSc in Biology and Chemistry at Maynooth University in 2013 and was awarded his PhD in Cancer Biology in 2018. 2020, he's the recipient of the Irish Association for Cancer Research Junior Investigator Award. So the floor is yours. Thank you.
Thank you. So I can just mime the slides. But yeah, just to say thanks, Jean, for the invitation to speak and really enjoying this conference. It's such a great opportunity to actually get to see people face to face again. We spend a lot of time on Zoom giving presentations, so it's really great to see everyone. Okay, thank you. So yeah, I'm just going to give a quick overview of what we do in Dublin City University and some of the projects that we're working on at the moment in ovarian cancer. So I'm based in the National Institute for Cellular Biotechnology in Dublin City University. It's on the north side of the city. So we're a research centre that's dedicated to translational cancer research and biopharmaceutical production and bioprocessing. So we've got strong links to local hospitals to gain samples for breast, gynecological, pancreatic, gastric, esophageal, and lung samples. And we're focused then on creating a culture collection bank. So it's mostly cell lines and then blood and tumor samples from patients. And we interrogate those for potential new targets. We've got advanced microscopy facilities. I'll show some of the work from that in a second. Proteomics, microRNA core facilities, and in vivo facilities as well. So I'm just going to go over one of the projects that we're looking at in ovarian cancer. We're investigating the combination of two targeted therapies in models of ovarian cancer. So one of these targeted therapies, neratinib, is a HER family inhibitor, and might be a little bit small, but there's the HER family has been targeted for over 2 decades now, very successfully. So there's a couple different modalities of targeting the HER family. The HER family is EGFR, HER2, HER3, and HER4. You can take an antibody-based approach, like cetuximab on EGFR, trastuzumab, pertuzumab on HER2, or an antibody-drug conjugate, where it's the monoclonal antibody trastuzumab with a cytotoxic payload conjugated to the Fc domain. Another approach has been using small molecule tyrosine kinase inhibitors. They bind to the inside of the kinase domain of the protein and inhibit its activity. These are orally available drugs, and we focus a lot of our work on that. We're investigating a lot of these targeted therapies in ovarian cancer at the moment, but I'm going to focus on the TKIs for this presentation. So lepantinib, neratinib, and dacatinib, these are all approved for HER2-positive breast cancer, but they haven't actually, there's very little data with a direct comparison between the different TKIs. They act slightly differently. Dacatinib is a selective HER2 inhibitor. Lepantinib is selective for EGFR and HER2, and both of these are reversible inhibitors. So they bind to the receptor and then can be released and bind again. Whereas neratinib is a pan-HER inhibitor, so it binds to all of the kinase active HER family, and it's an irreversible inhibitor, so it sticks to the receptor and causes internalization. can I move on? Yeah. So obviously these have been very successful clinically, but we wanted to compare in vitro to see which of the three small molecule TKIs was best across cancer types at inhibiting proliferation. So this was done in collaboration with Puma Biotechnology and the Netherlands Translational Research Center. So we looked at neratinib, lapatinib, and tucatinib across 115 different cancer cell lines, incorporating 25 different cancer types. And kind of what we were expecting to see was that neratinib, their irreversible pan inhibitor, would be best, was best. So not only was it effective in breast cancer, it was actually cancer agnostic. It was effective across different cancer types. And although our numbers here for some of our cancers are a little bit small, we've cervical, uterus, and ovarian as well, are one of the most sensitive cancer types. My work is focused on breast cancer for my PhD in the first 2 years as a postdoc. So I was looking to see how can we improve these HER2 targeted therapies, and the way that we discovered this was with a certain combination therapy. So we looked to repurpose approved drugs. So all the drugs that we're trying to select are already used in the clinic for different indications. So we chose dasatinib. Dasatinib is a multi-kinase inhibitor. It's known for inhibiting c-Abl in leukemia, but it hits a myriad of different kinases. And for this work, the SERC inhibition was most interesting. So what we found, to sum up 6 years of work in a tiny corner of a slide, was that the combination was really effective in HER2-positive breast cancer, in triple-negative breast cancer, and even in treatment-resistant HER2-positive breast cancer. So we found that although neratinib resistance emerges in our HER2-positive breast cancer model, the combination prevents this from occurring, and it's a prolonged effect both in vivo and in vitro and in vivo. We want to see, can this also be effective in ovarian cancer, particularly in epithelial ovarian cancer. The idea of targeting HER2 is not a new idea. In 1989, Dennis Slayman found that approximately 20% of the ovarian cancer samples that they looked at were positive for HER2. They had HER2 amplification. We now know the real numbers, probably about 5 to 8% have HER2 amplification. But I think there is scope for further investigation of the HER family in ovarian cancer. The clinical investigation has been quite limited, and the in vitro work is needed to justify the clinical work. So that's where we come in. So first, to actually take some of our cell line models and make sure that our targets are being expressed. So we have HER2, EGFR, HER3, and HER4 are her family members. So we've 6 ovarian cancer samples here, cell lines here, and we can see that the targets for neratinib are all commonly expressed in our cell lines, and even the cell lines that don't express one particular member of the family will have overexpression of another. So it's showing that there is potential there for targeting with neratinib. A key thing to look out for as well is that the downstream signaling is also activated, that we don't just have activation of EGFR, we want to see AKT and MAP kinase activation. And we can see that there's constitutive activation of AKT and ERK here. And then when it comes to dasatinib, our targeting of SERC, we have activated SERC expressed in all of our cell lines. So this was a good thing to see before starting our drug investigation. We also want to look in 2D and 3D. So we can see that in 2D and in 3D, we have either continued expression and activation of the HER family pathway, or an actual increase in activation. So that's translating our 2D to 3D culture. So then actually looking at the response to the drugs themselves. So taking our panel, we carried out 5-day proliferation assays, and we calculated our EC50 values. These are all nanomolar concentration. So all of our cell lines showed good sensitivity at nanomolar concentrations to neratinib. All of the work that we do, we want to keep it at peak plasma concentrations, what's actually achievable in patients. So the peak plasma concentration is about 150 nanomolar in neratinib. So although there's sensitivity here, it's sensitivity that can be improved upon. Funny enough, dasatinib, which we didn't expect to work very well in their cell lines. There was one clinical study of dasatinib in ovarian cancer about 10 years ago, and there was little single agent activity. Was very effective in vitro, very low nanomolar concentrations, and 6 to 7 were highly sensitive. And we found that there was no correlation between the drug response and the protein expression that was previously shown. This is most likely due to the fact that both of these drugs are multi-kinase inhibitors. They're hitting a lot of things in the cell. It's not going to be one particular marker of response. So we're expanding on this at the moment to try and have a panel of markers of response. So the goal here was to see the effect of combining the two together. So we assessed this by matrix synergy assays. The color didn't come across great here. So the way that we do this is we plate the cells and treat with one drug with increasing concentrations from left to right. So in this case, neratinib concentrations increase from left to right here. And then dasatinib concentrations increase from top to bottom. So that means that this line here is neratinib alone, this line here is dasatinib alone, and everything in that yellow box are different concentrations of the two drugs. So for 56% growth here, that's 8 nanomolar neratinib and 1.6 nanomolar dasatinib. The advantage of this type of assay is that it gives you a much better indication of potential synergy and also potential antagonism. These sort of small molecules at very low concentrations can actually activate the target, and that's been seen before with some of these TKIs. So it was important as well to make sure that there was an antagonism. So we take our percentage growth, use ComBenefit software, and we get our low synergy scores. The higher the number, the more synergistic. If it's a negative number, it's antagonistic. And importantly for here, if you see blue, it's synergy. If you see green, it's additivity, and red is antagonism. So this is in one of our cell lines. This is the SKOV3 HER2 amplified cell line. We carried this out in our full panel, and you can see that 6 of the 7 cell lines had synergy with the combination. The other important finding was that there was no antagonism seen across any of the cell lines, including the cell line that actually didn't have any enhancement with neratinib and dasatinib. This is something we've seen in endometrial cancer cell lines as well, and also across breast, gastric, and lung cancer. So we're really excited about this combination. And the other important thing about this result was that, again, these are all at low nanomolar concentrations. These are achievable in patients. And what we hope then is that we could combine lower dosage of these two drugs for patients. So the previous slide was looking at 2D growth. So we wanted to see, can we also see this effect in 3D? So we use a system called the IncuSight S3 system. It's a kinetic live cell imaging system. So it's a very fancy advanced microscope that sits inside of an incubator, and we can control when images are taken and add different fluorescent dyes. So we can measure a lot of different things with spheroid, proliferation, apoptosis, antibody internalization. There's a lot of different functions to this. So this is one of our most important machines in the building. And just taking one of our cell lines. So these are the AVCAR3 cells with spheroid growth. The darkest blue is untreated, as we expect, continues to grow for 5 days. Neratinib is a bit less effective as a single agent in the AVCAR3s in spheroid growth. dasatinib, and then our synergistic effect with the combination. So it's not just inhibiting, we get this haze of dead cells around our spheroid. So it's killing the cells as well. But we needed evidence for that. So we've looked at these drugs, mostly in breast cancer, but in different cancer types, and there's several different mechanisms of action. So what we found in breast cancer was that HER2-targeted TKIs can not just cause cell death by apoptosis, but can also induce senescence. That's important for arresting the cell's growth, but then when the drug is taken away, those cells can potentially grow again. And then with a slightly different combination, another pan-HER inhibitor, afatinib, combined with dasatinib, we looked at this in triple-negative breast cancer, and we found that rather than apoptosis, it was purely a cell cycle arrest phenotype that was being caused by the combination. So our question was neratinib and dasatinib in ovarian cancer cytostatic or cytotoxic? And the overwhelming answer was cytotoxic. There was a really strong induction of apoptosis, which I will show you in a second. Sorry, just building up suspense. So I look for, don't think the videos are playing. It's okay, I can describe it to you anyway. So again, using our Incucyte system, we analyzed the growth of the cells again. So the graph on the top left there is just looking at our, we'll get there. No? So the top left there is just confluence. So just seeing what we saw before that we're inhibiting growth. This is kind of what we expected. Yeah, that's okay. No worries. For this one was 2D. Yeah, but we can do it in 3D as well. Thanks. Cool. It doesn't like the videos. Okay, I can kind of just, if you want to skip on to the next slide, I'll just describe it because it might crash it again. I just realized the next slide also has some videos, so. So just to describe the previous slide for a second, could you go up one on outside PowerPoint? Oh, cool. Oh, it's working now. Okay, cool. Okay, so what we can see here is our 2D growth of the SKOV3 cells in this case. Top left, again, we're just seeing neratinib and dasatinib single agent work, and we get enhancement with the combination. This is at 150 nanomolar concentration, so keeping that peak plasma concentration. The important result is the graph there at the bottom left. So this is caspase 3/7 activation, so it's a marker of apoptosis induction. We do see the combination undersells the single agent effect. We do see an induction of apoptosis with our single agent, but we've an incredible increase in caspase 3/7 activation even after 2 days of treatment. So you can see that here, our cells becoming confluent in the control. Our pink here is the caspase 3/7 activation. So relative to the cell population, we can see this induction of apoptosis. Okay. And then to find out if this effect can also have anti-migratory impact, we looked again using our Incucyte wound scratch assay. So we took our 3 hour time points again with treatment, and found that the cells are migratory and they close pretty quickly for ovarian cancer models. Dasatinib has a pretty good effect of inhibiting that, but the combination works best. So in conclusion then really, we found the single agent is quite effective across the cell line panel, but there's extremely strong synergy in 6 of the 7 models. The combination induced apoptosis and prevented cell migration. What's currently going on is we're starting the in vivo study in the next couple of weeks, and we're doing reverse phase protein array interrogation. We've done this for our breast cancer models, but we're doing a large 490 analyte analysis because these are multi-kinase inhibitors. We want to see all potential pathways that could be affected with the combination, and we're also planning on doing investigation in organoid models as well. So thank you, and just like to say as well, open for collaboration. So any of the technologies here, if you molecules that you're interested in, or I can help with anything, talk to me. Thanks. Yeah, no worries. Well, I hope they don't crash this time. The breast cancer.
Were those skid mice with cell
Yes,
line?
yes, it was nude mice, not skid.
Okay, nude mice and with the injection of the cell line.
Yes, so with the breast cancer model, that was orthotopic into the mammary fat pad. With the ovarian cancer cell line, it's going to be on the flank because.
So my interest always is, you mentioned that you're using plasma concentrations similar to those used in patients.
Mm-hmm.
However, whenever we do this kind of research, we always use either proliferating lymphocytes or proliferating cord cells as a natural control. I always am not impressed by things where you say it's killing everything. Yes, show me normal cells.
We've actually, I don't have it in the slide here, but we've looked at live PBMCs and normal breast cancer, normal breast immortalized cells. We've done a lot of work with those as well, and we don't see.
So proliferating PBMCs, when you stimulate the lymphocytes to proliferate.
Yeah.
You stimulate them.
And we don't get.
And you don't see any toxicity.
No.
That's weird, considering you have a multi-kinase inhibitor.
Yeah, we're not getting the inhibition with it. There's some inhibition, but it's not similar to what we see in breast cancer.
That's extremely interesting. The other thing, which is I liked a lot, I think it's the slide after this or before this, where you have the panels of combinatorial work. Yes, over here.
Yeah.
So if you see the ones where you have the darker purples and blues, you seem to be having it happening at a high dosage of the neratinib, but at a low dosage of the dasatinib. Do you have any idea why? Do you have any suspicions or hypotheses why that's happening?
So I think it's partially due as well to actually having the effect with neratinib. So you can see the percentage growth here. So at 200 nanomolar for this cell line, there's basically no growth inhibition with neratinib. It's only above 200 that you're actually getting the inhibition. Looking at the kinome of the two drugs, neratinib, as I said, is multi-kinase as well, but it's hitting its main targets at very low nanomolar. So I don't think it's an off-target effect that's occurring. I think it's actually that you're just getting more of the inhibition at that point.
Why wouldn't you expect that the higher dose of dasatinib you'd get better inhibition?
Say it one more time, sorry.
If I'm misunderstanding, I might be misunderstanding. If you look at where the two purple blocks are on number 3, right?
Yeah.
Wouldn't one expect that if one was to go further down where you have a higher dose of dasatinib, you'd get a better inhibition rather than a worse one?
Yeah, I think that's partially it's your scope of getting more inhibition. So going from your 24, 25% down to 10, it is synergy, but it's not getting the same wipeout as you're seeing with the slightly lower concentration.
So you have no hypothesis why that's happening?
No,
okay.
No.
It's very interesting.
That's
why I wanted to say. You have a similar one in the one down there.
Yeah.
Again, it's working at very low doses.
And we see this across the cancer types as well. And we're looking at particular knock-in cell lines or cell lines with particular mutations to try and see for example, like the PO1s here are pre-treatment, PO4 is after chemotherapy treatment. So we're trying to look at those sort of match models to see what's shifting and potentially can we get a marker of resistance or a marker response.
Thank you.
Thanks.
kind of a follow-up actually, because you hinted at it in your presentation about resistance to the TKIs, especially in leukemias. That's a huge problem, right? And you kind of, have you looked at longer trajectories and see whether you get resistance?
To the combination?
To combination, yes.
The longest we've looked at is just with the in vivo study. So that was 10 weeks, so 70 days with treatment. And I took those tumors and tried to grow them afterwards, and they were still sensitive to the combination with treatment. That was in our 1954 breast cancer model. We haven't yet looked at resistant single agent statinib. We've looked at single agent neratinib resistance and kind of how it overcomes with that. But we haven't looked at the mechanisms of combinatorial resistance yet.
And I know it's done in chemotherapy. I'm not a medic, but has anyone looked at tyrosine kinase inhibitors in terms of metronomic dosing? So rather than combination, you use one and then you just add every now and again the others.
I haven't seen it with any of these sort of drugs. So both of these drugs are given daily orally for patients. No, I've never seen that sort of situation. There has been kind of like we had a phase 1 trial last year with combining neratinib with some monoclonal antibodies, and there was that sort of phasing, but not for the TKIs. That would be would be interesting to see even in our in vitro models if we do a pre-treatment single agent and then add in afterwards. Yeah. Thanks.
Thank you.
Yeah, so we use a
Okay, so I need to repeat. What system do you use for 3D culture? Is it the hanging drop or the non-adherent plastic or something else?
Yeah, so we kind of take a combination approach. The assay that I showed here was taking non-adherent U-shaped wells and growing single spheroids. We also do actual matrix gel suspension. We can use our IncuSite for that too. That's I kind of find is more like a 3D colony forming assay really. I wanted to see actually taking a larger cell mass, could we get that inhibition? So yeah, to be honest, 2 approaches really. And we can look at organoids with this system as well, and that uses the multispheroid based approach too. Yeah, perfect. Thank you.
Very good. So we move on to our next presentation, which is by Dr. Mariela Vasileva-Slaveva from Bulgaria. So Mariela, she's a surgeon, she's a medical doctor and a surgeon at the Department of Surgery in Sofia, Bulgaria. And she also works at the Research Institute at the Medical University of Pleven in Bulgaria. And she's a board member on the Bulgarian Breast Cancer Association. She has a special interest in breast cancer surgery, in biobanking, patient-reported outcomes, and education in oncology. So she's also had fellowships at the University of Innsbruck in Austria, and she had won the Carroll Knowledge Foundation Award for Entrepreneur in Science in 2021. So this is just a brief summary of all that you've been doing. So thank you. The floor is yours.
So hello everybody, and thank you for the opportunity, and special thanks to Jean for the invitation. I would like to speak about the news resources of rare gynecological cancer in Eastern Europe. And I'll first start with data from our Bulgarian National Cancer Registry. Actually, Bulgaria is one of the very few countries in Eastern Europe that has a national cancer registry. Registration of cancer patients in Bulgaria started in 1992. And we have a national electronic database since 1993, 1952 and 1993. Previously, it was very well organized and structured. We had 13 regional cancer registries, and each of them had to send their cases to the national database. But in the recent years, the law has changed. Now every hospital has, but it's not obligatory, to send their data for the national registry. And of course, this has a lot of troubles because not all, especially private hospitals, are not sending their data, and the data in the national registry is decreasing annually. So what I did for this presentation, so currently in our database, there are almost 1 million cases. And of those 1 million cases, over 10%, 100,000 are gynecological cancers. Now, from these gynecological cancers, without detailed histology, like I couldn't say if they are rare cancer or not rare cancers, are 10%. And I took very literally and by heart the table from our publication that we have to see which are rare cancers and which are not. So these are over 12%. And so there are a lot of limitations in this study. So I am the first limitation because I'm a surgeon, I'm not a gynecologist. And I took the liberty to do some statistics, but I'm not a statistician. So but the point of this is to show that actually this data is useful, and yes, it can be very much used, and it's worth it to collect it. That I should not convince you in this. So this is the table that you have, and these are all cancer cases that are registered in our registry for the whole time ever, and the percentage. And I discussed these cases one by one. So you can see here the total number of cases, and the rare group, the rare number is below. And vulva and fallopian tubes I consider all rare. Okay, yes, now we can. So the vulva vagina, they're all rare cancers. So I did the Kaplan-Meier. It's not adjusted to anything, no stage, nothing, but it's 5 year, and here's the 5-year survival rate. And I in order of decreasing survival of these cancers. So you can see here for vulva, for cervical cancer, I will speak a lot about cervical cancer in my presentation later on, but you can see here that adenocarcinoma, for example, has better survival than carcinosarcoma. Maybe it's not surprising. The uterine body, again, here the sarcoma has this is only rare cancers and only cases that have more than 10 cases in a group to do the Kaplan-Meier. Otherwise, it was not meaningful. Fallopian tubes that are all rare, but you see here I had the same problem, like they're all rare, but for over 100 cases, I couldn't see exactly in which type they go. So it's only one only one type here, adenocarcinoma. Yes. And for the ovaries, you can see here I did the survival in each group, so decreasing survival in each group. So in the epithelial, which has the best survival, low-grade serous carcinoma, in the, for example, in germ cell, dysgerminoma has the best, and so on and so on. Now, I wanted to discuss the curious case of cancer of the uterine cervix, but while I was doing this presentation, I realized this is not the curious case. Actually, the curious case are the other gynecological cancers, because for cancer of the uterine cervix, we know why it's happening, what it is happening. And what it is actually is that in Bulgaria, we have 6 times more cancer of the uterine cervix than in Malta. We have 3 times more cancer of the uterine cervix than the European mean. And this is, we know the reason. So here I wanted to show that in Malta, you know, it's a very, like, it can be, the whole cervical cancers can be considered rare. But in Bulgaria, we have only almost 20 cases per 100,000, and we are 4th. This is Globocan, European Agency for Research on Cancer. We are 4th on the highest rates of cancer of the uterine cervix. And there are 2 main reasons for this. So one is that we don't have vaccination, and we have a lot of HPV. And here are again from there Results for cancer of the uterine cervix, and you can see here Romania, our colleagues in Bulgaria, and the whole Eastern Europe. And here is the distribution of HPV among women with normal cytology. So it's getting even better. We don't have data here, but still, the highest rate of HPV is there. And also here you can see in different regions of Europe, like in Eastern Europe, we have in every age the higher rate of HPV in women with normal cytology. So this is one reason. And the second reason is that Bulgaria is the only country in European Union that doesn't have a screening program for cervical cancer. And you can see here the different countries are in different stages, and this is quite old, 2016, but it haven't changed by now. Bulgaria still doesn't have a national screening program. It's the only country that doesn't have. So these are the outcomes of from Eurostat. The population covered by screening in different countries, and you can see here countries around like Czech Republic and Slovenia, and the green is European mean. And I even couldn't find this program who covered 40% for 1 year of the population, because I couldn't find it. I really looked for it, and how Eurostat get this data, I don't know, but at least this was the only attempt that we had. And so we took cases only cervical cancer 2015, 2020, and you can see here this is in different stage, 1A, 1B, 2A. It's not surprising, of course, this. And this is the mean age diagnosis. So, of course, if we had screening program, we could get a lot of cases in an earlier stage, and it's obvious, and you all know it. So let's hope. Now, Bulgaria actually, at the moment, since the European directive, all European countries should have an anti-cancer plan. So we are now developing it, and they say there should be 3 cancers, and cancer of the cervix is one of them, that should implement screening. But let's see. Okay, so actually the other cancers, this one was easy to explain why, but for the other cancers, I only can guess, or even not to guess. This is cancer of the uterus. So you see Bulgaria is on the 3rd place in cancer of the uterus. And yes, in Switzerland, it's not rare, yes, but still quite small numbers. And this is ovarian cancer. And for the uterus, I discussed with colleagues, maybe we are obese, diabetic, and stuff like this. But for ovarian cancer, they couldn't think of a reason why it's so high. And you see here that in Kuwait, if in Netherlands, let's say, because I don't know if we can trust, knowing our registry, I don't know if we can trust other registries, but still maybe Netherlands we can trust. And there the whole ovarian cancer is rare. And you see Bulgaria here is for sure not rare. And so here is again uterus and the ovaries, and you see, and here upstairs is where there are biobanks. So actually we have a lot of resources that are unused. I don't know if something to be proud of to have a lot of cancer, but maybe we can use it and actually put it in a biobank. So this was our project that we did last year. It's a first Bulgarian virtual cancer biobank. It's now starting. It's not ready yet. It hasn't launched. But our idea is to make a software where we can keep the data virtually and not only to collect paraffin tissues at the moment, because otherwise it would be very expensive. But we can start from this, and every institution can has its own database, but all the databases in one. And we actually started already with one university, Medical University of Pleven, and we I think we're very close to do it in other 2. We're now in process of developing the software, and we always ask why people should decide to go in our biobank, because we happily live without biobanking by now. So our idea is that actually this will give every doctor, I'm sure it's in Bulgarian, so that's why it's not so important what's written, but it will give the opportunity to every doctor to have his own database. Every doctor who participates in the biobank can have structure his data so he can use it. And in Bulgaria now we have a transition also in education, medical education. So actually people will be, I don't know to say forced to do research, but yes, if they want to have outcomes. So they would like to do it. They do it anyway. When I was student, I did it on paper. So now it's much better if they just collect while they're on a daily basis on their work, they just have this database ready for them. And yes, we have one award with this project. In 2021, Entrepreneurs in Science. And also regarding this, I will appreciate everybody's help and participation with Jean. We're making a virtual mobility on feasibility, regulations, and sustainability of virtual banks in Eastern Europe next month. So everyone are invited to join. And that's it. Thank you very much for your attention.
I will just make a comment if it's okay. So I think this is a case in point where, I mean, COVID in a way has helped because it has made us a bit more courageous in going virtually, and even COST have launched the virtual mobility grants. And it's one way of, I mean, reaching out, especially, you know, when you have busy people and who might not be able to travel or meet regularly, or, you know, like in this case, we're having meetings every Thursday. So, you know, so you can still continue with your work at home and still collaborate and reach out. So, and again, that's the beauty of a virtual mobility, because you could have, you know, people from all over Europe or the world contributing. So would like to see you there. And what we can do is I'll pass the link round, you know, like I'll do with the surveys. So whoever wants to chip in, like Nikolai, I mean, anyone with an experience in biobanking would be, you know, I think Bridget is involved, you're involved, I think, in this, yes. So we'll put, and Sharon, of course, yes, exactly. So thank you, Mariella, thank you so much.
I was going to ask a question.
Oh, sorry, I didn't see you. Sorry.
Okay.
Scusa.
Mariella, thank you very much for your work. It's obvious that your work is very heroic, actually. No, I don't. I mean it completely. I wanted to ask, what is the health service like in Bulgaria? Is it a national health service? Is it paid by taxes? Is it an insurance? What's the system?
We have a national insurance fund, which doesn't cover all of the expenses, and we are the country with the highest rate of out-of-pocket patients from payment from patients.
Okay, so I'd like to make a suggestion to Jean and to yourself that the cost section should, in my opinion, take this upon themselves to say, if we are ready to put money together from all of Europe to buy COVID vaccines for all of Europe, including our small country of Malta, which benefited a lot for this, I think it's about time that Bulgaria doesn't remain an exception on this about a cancer which we know how to treat and how to prevent, and they have not got the money or whatever to buy the vaccines and the screening programs which are needed. And I think GYNOCARE should make this a #1 priority, and to use Metsola as well, our Maltese president, who's a female and who's making a lot of fuss about equality in women, which she should, to make this a point, because it's idiotic that we have something which we know how to treat, we know how to prevent, and we are letting people die because of it.
It's a very good point.
I'm not sure. I'm sorry. It's not like I don't take money. I will for sure. I'm not sure it's a money problem, because now with this European, as far as I know, now the European Union will give money for this anti-cancer plan. The thing is that how this money will be then used in Bulgaria, and then I don't know how can we influence this process, honestly.
But that's part of why the EU is here, so we should make use of it.
I mean, in fact, I mean, you read my mind, Pierre, because I mean, as you know, the president of the European Union at the moment is Maltese. I mean, she's female, and we know that she's an advocate in health. I mean, among other things. So I think one way maybe, and I'll be mentioning this even in the MC meeting. One way is that if there's a meeting held in Bulgaria, that we get it, get the press on, etc., in order to exert influence. And of course, you know, I mean, Bulgaria have been very active. Mariella and her colleague Angel, they have been very active and they've participated. So we, you know, so that your work shows and the impact shows, I think it would make sense also to, you know, help with the promotion there.
Yeah,
for sure. So it's one thing that we should get together and help out for sure.
Is the program being formed, written, organized? Is it a problem because the program has not been written or because?
but they say, well, it's really on a day by day it changed. Now the problem, I don't know, it's in your countries like this, but our oncologists, they fight a lot. There are two schools of oncologists now in Bulgaria who even succeed to fight in front of the European Commission who to do the anti-cancer plan. So and they one is say yes, I'm doing it every day, and the other says yes, of course I do it every day. So finally someone is doing it. What is written there, I don't know.
Is there a public health department? This should be public health. I mean, ours, I think, the registry people, public health people. It's more. You can maybe speak. It's more or organized on a public health level rather than by the clinicians or whatever. Yes.
Definitely, with your data in hand, the data can be passed to policymakers in your country, and sort of they would be forced to work upon it. Another thing is that this data that has been collected in your country is available in these international databases, including European databases. And there are also currently studies that show the outcome of these cancers. In particular, for example, we have results from GYNOCARE version 6, or the sixth edition, and Concord 3. If a particular country shows a particularly poor outcome in terms of age-adjusted survival after 5 years, this is available even on public websites. And it is even brought to the attention of the European Commission. I am aware at the moment that there is consultation across Europe on these cancer plans, and there have been issued guidelines on screening. Eventually, this screening will be made mandatory. They are currently discussing the best way of putting it forward, including the ages that should be screened, the types of cancers that we should screen for, the frequency, and the best methods for screening. It is very useful that we collect data, because this data is available for everyone to see on these international databases, and sort of it is brought to the attention of the European Commission, and countries who lag behind sort of will be forced to take action. At the moment, all the countries are also participating in research about cancer, and this is being carried out by OECD. At the moment, OECD is writing a publication and describing the cancer profile of 29 countries, including most of the European countries. And here in this cancer profile, there will be anything from the epidemiology and also statistics related to outcome, statistics related to the services, including, I mean, I have participated in one of these focus groups just a week ago. We were being asked when this screening program was started, for which cancers the screening is present, the frequency, the coverage rate of the population, and everything. So eventually, even this data will be available for everyone to see. This major data collection initiative started as part of a smaller project called the formation of Cancer Inequalities Registry. This will describe cancer inequalities related to everything, to outcome, to services, to incidence rates across Europe. There is already like a form of mapping or ranking in this Cancer Inequalities Registry with a number of indicators and variables that have been used to describe the rankings in the countries. Not exactly rankings. I mean, there is a check mark for what is being done in the country or not. And also, eventually, this Cancer Inequalities Registry has already collaborated with OECD, so that OECD will first produce these 29 country profiles. The interviews are currently in place, and the publication is expected to be in November 2022. So we will see something written about each country. We are all anxiously waiting on the reports of our countries. Eventually, OECD and this working group, the Cancer Inequalities Registry Working Group, will also produce an overarching report. It will analyze the 29 reports, all the data that it has in hand, and it will produce an overarching report. I am aware also that in Europe there will soon be a joint action on inequalities in cancer. The call for participation has already been issued, and for example, I have been nominated as a representative of the registry to take care of this joint action. So there is more work in the area of inequalities related to cancer across whole Europe. These collaborations, as we were saying, are very important so that we know the status of our countries, we can compare ourselves to other countries, and see that no one is left lagging behind.
Thank you very much. I hope we can do something of this. We'll see what will do our anti-cancer plan and how it will be implemented, because by now not really successful, but we'll see. Thank you very much.
Mariela, thank you very much for your presentation. Very important. I think that the situation is very difficult in Bulgaria because the results of the GYNOCARE project and the RareCare Net were to abolish the cancer registry, population-based cancer registry, because now, yes, they have a sort of collection, registration of data, but just to estimate the cost of drugs. This was an explanation. So there is nothing after the GYNOCARE 6, because that's the situation. We had two meetings in Sofia because of the RareCare Net project, showing the results and inviting the public health people, and one for rare cancer and another for childhood cancers, because also for childhood cancer there are treatment, but the survival is very, very low. And I think that the private treatment is ongoing strongly in Bulgaria, and the public health, the access to treatment is very that. So I think that we need to write a paper on a scientific, just to describe this situation and profit of the European cancer plan that was. That is important.
If I can say, with Angel, we have started a paper like this describing this, but we haven't finished. We will be very happy if you someone can join and help us. Yes, because I'm struggling with the statistics actually. So
yes. Yes, and we've got the expertise. You've got the statisticians. You've got the RareCareNet. Yeah, the European networks. So
yes, it will be great. I'm ready to share.
Yes, and it will probably alleviate a bit of your burden on you as an individual in Bulgaria. So because you've got the support of a much bigger network from Europe. So you can't be targeted as saying, "Oh, what are you doing?" You have the support of the collaboration.
Yes, it will be great. I highly appreciate it. So great. Thank you.
And,
with reference to the issue of HPV vaccination, there is pressure from the EU to extend HPV vaccination even to boys who are 12 years old. Another thing that might need to be pointed out, but I'm not the best person to talk about it. I'm not a pathologist. Is that the latest version of the World Health Organization Blue Book, which defines the histologies, has included some new categories of these rare gynae tumors that are dividing these rare gynae tumors, some parts of them into HPV associated and HPV not associated.
Yes,
in fact, I remember that in your survey.
Professor Simon Harrington, who is the author of the Blue Book, is actually in the network. So maybe he would be one to speak at one of these June sessions, if that would be ideal, just to target. Because you have to ask him early enough because he's very busy. That's why I'm saying. So maybe you should get in touch and find out when he's available to give you the talk on one of which of these June meetings you are having.
Yes.
He's actually part of the GYNOCARE. He used to be our external examiner in Malta. So, and we invited him and he's accepted. So, exactly, he's lead of the, so maybe that is a good way of finding out exactly what you should be collecting from him, maybe.
Thank you very much. It was great. Thank you.
Seen the interest and not just the interest, but the will to help. So it was, excellent, and we'll definitely continue working on this. I mean, this is the whole spirit. So thank you very much, Mariela. Now, I would like to present our next speaker, who is Dr. Paul Kubelak from Romania. So Dr. Paul, he's a medical oncologist working at the Cancer Institute Ion Chiricuta in Cluj-Napoca in Romania. He has a PhD in medical sciences focused on evolutionary perspectives in ovarian cancer. Has numerous publications and he has been awarded a fellowship at the Gustave Roussy in Paris. He, of course, has a special interest in gynae-oncology, focusing on ovarian cancer. So we're very happy to hear your talk, Paul. Thank you.
Thank you for the kind presentation. And I would like to make a brief overview of what we have done from a research perspective in ovarian cancer in our institute, which is located in Cluj-Napoca. It's in the northwestern part of the country. We have 3 large cancer institutes in Romania, one in Bucharest, one in Cluj, one in Iași. Of course, there are also other smaller centers and private ones developing, but we have patients coming and they are treated at our cancer center from across the country, not only from the western part. And we also have basically the single functioning cancer registry that is also reporting data to GYNOCARE and to Lyon. And we are also members of the GYNOCARE network since last year. So talking about ovarian cancer, we know it is a silent killer. We have non-specific symptoms. We had a video and a presentation yesterday about it. And unfortunately, in comparison with cervical cancer, we don't really have a screening program that works. I have put here two trials that have been done. First is the UK collaborative trial on ovarian cancer screening, and the second one, the PLCO trial, and none of them managed to show a reduction in mortality in the overall population. Of course, this is not valid for BRCA carriers, for which we know that it is recommended to do prophylactic surgery after a childbearing age. And incidence, it's considered a rare cancer, ovarian cancer, but we can see we have a large incidence, around 300 cases in 2020 reported, growing since the last report in 2018. And what's very frappant is the fact that mortality is very high. It's basically the deadliest gynecologic cancer, and we can see that it affects both high income countries and low income countries with the same incidence and similar mortality rates. And this is a presentation in a few images of what our colleagues from the surgical department are usually treating. We see that usually at diagnosis, we have diffuse mesenteric involvement cases that are not suitable for primary debulking surgery. We have cases that end up with bowel obstruction either at diagnosis due to late symptoms or through the course of chemotherapy. We have cases that have diffuse intra-abdominal relationships that make surgery very hard, but we have a very dedicated surgical team that can perform in-block resections of the pelvis, in-block resections from the GI tract, retroperitoneal surgery. And it's important to know that in ovarian cancer, if you want to do surgery, it must be R0. So this means no complete residual. This is the most important prognostic factor. And this is why this is a short introduction towards the fact that sometimes we use neoadjuvant chemotherapy to downstage the disease so we can make it operable. Since 2010, we have been working on various publications in ovarian cancer, and I want to make a brief overview of what we have done and maybe share some ideas. And if you are interested in research projects or collaborations, we are very open towards it. So I have structured them regarding their objective. First, this is a paper we did 2 years ago regarding diagnostics. So I said we don't have a working screening program. Diagnostic is very hard to achieve. Basically, we all know that diagnostic is a pathological result in ovarian cancer. And we have a ROMA, the ROMA algorithm that can give us a sensibility and a specificity regarding the possible malignant or benign characteristics of an ovarian, of an adnexal tumor mass. And we have developed this OncoVarian DX model where besides the classical HA4 and CA1 to 5, we have added other markers. And from our preliminary study, we have seen that this model demonstrated good sensibility compared to ROMA and could be useful as a marker for subsequent development in the pre-surgical diagnosis. Another study we did around 3 years ago regarding diagnostics is the surface enhanced Raman scattering based differential diagnosis between multiple solid malignancies. So this is a technique in which we measure the Raman shift using serum samples from we used the control samples and ovarian cancer samples, and we saw a very good concurrence between a predicted risk of diagnosis and reference sample. And in ovarian cancer, we had an accuracy around 80%. So maybe if we design something that has that can combine, for example, Raman and multiple tumor markers, maybe we can enhance our preoperative diagnostics. Now, moving towards prognostic markers, we have done several studies, also many of them focused on the tissue microenvironment. And this is one of our first studies we did. It was the role of inhibitor of DNA binding 1 protein and angiogenesis in ovarian cancer. And we saw a very interesting association between this protein and microvessel density, which was significantly correlated. It was not correlated with other outcomes, but this we considered an important step, because in ovarian cancer, angiogenesis has been largely debated. We also studied other markers regarding tissue microenvironment, for example, CDCP1, which is a CAP domain containing protein, and ADAM12, a disintegrin, and a metalloproteinase. These are markers from the tumor microenvironment, from the stromal tissue. And we saw that both of them, their immunohistochemical expression was correlated with survival. So if we had high expression of both CDCP1 and ADAM12, we had a poorer prognostic for our patients. Going further, we also looked at nestin and CD133, which are regarded as putative markers of cancer stem cells, which are related to prognosis in various malignancies. And we also investigated them in a research article we did. And we saw that they are correlated with progression-free survival and overall survival, especially nestin. We didn't get positive values for CD133, but expression was correlated with, so high expression was correlated with worse survival. Also, we looked at CD146, which is an cell adhesion molecule, also capable to modulate the activity of endothelial cells. And we also got positive results from this study, and showing that positive samples also had a worse prognosis. We also tried to do a lot of research with the Tumor Bank Ovarian Consortium, which is located in Charité in Berlin, Germany. And we sent samples, and there was a joint analysis of samples of especially long-term survivors in ovarian cancer. When I mean long-term survivor, we usually define these as patients that live more than 5 years. Generally speaking, patients with ovarian cancer have survival less than 5 years. So analyzing this data, the collaborative group showed that overexpression of vascular endothelial growth factor receptor 2 was correlated with long-term progression-free survival, which was independent of patient age, FIGO stage, residual tumor, and VEGF-A expression. And these were all patients that did not receive anti-angiogenic treatment. And regarding prognostic, we also thought that it would be interesting to study the role of the immune system. And we did the study on toll-like receptors, which are important in the innate immune response. And we saw that, for example, in early stage cancers, toll-like receptor 9 was correlated with survival probability with overall survival. And in advanced cases, toll-like receptor 7 was also correlated with survival. And what was also interesting to see in the study, we did two markers, CD68 and CD163, as markers of macrophage M1, M2 polarization. And we saw that if there was a trend towards M1 polarization, which brings better survival, patients had a longer platinum-free interval. Some recent research we did also on prognostic factors. We did a comprehensive analysis of key genes related to hippo signaling. This pathway signaling has been very conserved from an evolutionary point of view. We can also find it in flies and in Drosophila, and similar protein members are also found in humans. And this pathway regulates organ size and growth. And we saw that we did an online analysis of public available data, and we also did a subsequent step of validating our results. And we found that different expression, I will not go into this details from time concerns. We saw that it's prognostic if we interrogate the hippo signaling pathway. Doing also collaborative research in this multi-center analysis with centers from Berlin, Hamburg, Innsbruck, Leuven, and Rome. We tried to define a clinical profile for long-term survivors because we are lacking prognostic markers for long-term survivals. And we also wanted to see from a clinical point of view what is relevant. And we saw that recurrence, ascites, and tumor involvement in the upper abdomen are detrimental factors for long-term survivals. We also saw that platinum sensitivity constitutes a favorable prognostic for long-term survival, and only combinations of several factors can determine this long-term survival. We thought it's also important to look at things also from a quality of life perspective. And it was this international no-go ENGOT trial called Expression 3, where patients were handed questionnaires. And we extracted data for the Romanian population from this questionnaire. These data were aggregated. So all questions were structured into using structural equation modeling. They were structured in 4 categories. So physician's evaluation by the patient, results of therapy, need for changes in treatment, and patient's desire to be informed. And we saw that there was a significant positive effect of a physician's evaluation by the patient on results of therapy. So if the patient considered his physician highly capable, then the results of therapy were also influenced. But if the physician had a negative evaluation from his patient, the patient and this was significant, both of these relationships were significant. The patient felt needs to make changes in his treatment. And looking also at this expression 3 overall from all the data that was aggregated from close to 2,000 patients, we saw that ovarian cancer patients have a high need of information of all details regarding treatment options, which is irrespective of their cultural background, of the stage of their disease, or of their patient's age. So it's very important to develop efficient communication tools with ovarian cancer patients, given the protracted course of this disease. And expression 3 was had a follow-up, which was expression 4. This was also a questionnaire-based study, which evaluated the maintenance phase of therapy in ovarian cancer, because we know we do a lot of maintenance nowadays after chemotherapy. And this study highlighted that patients were motivated to accept maintenance therapy with a longer duration. They mostly preferred to get pills and not IV treatment as maintenance, and they were willing to accept it. And still there is a gap between the efficacy of maintenance and their patients' expectation. As you can see, most of them wanted a delay of more than 6 months. Most of them were willing to do it until tumor progression, and strikingly, most of them wanted that this maintenance would offer a chance of cure. But actually, with maintenance, we only delay the mortality, but it's not really what patients expected. And we did a few surgery analyses regarding the complete resection rate we got on a retrospective analysis we got around 60% complete resection rate, and this was highly significantly associated with survival. This is as a primary debulking procedure. And we also did another study on secondary surgery in ovarian cancer patients. Secondary surgery in ovarian cancer is very debated. Until a couple of 2 years ago, there was a lot of debate if there is a role to do it or not. We saw in our retrospective analysis that if you do surgery and chemotherapy, patients would have a better survival, 52 months versus 40 months only for chemotherapy. Now, since this study, we have some results. The Desktop 3 trial was presented at ASCO. It showed improved survival, but only for patients with positive AGO score. GOG 213 showed no benefit in overall survival, but this was a trial that had a lot of Avastin maintenance, and this might have influenced the final outcome. The soccer trial was stopped due to poor accrual. And there's also the SOC1 trial, from which we don't really have final data yet. We also looked if it's important to do PET imaging in ovarian cancer. And in a small study we did, we showed that if you do PET CT, you are more likely to get a change in your therapy than doing simple CT. And this is important because, as I said, surgery should be done until there's no residual disease. If there is residual disease, then there's no really a role for doing surgery. And this is a project we like most. So, as I said in the beginning, you can do surgery prior to chemotherapy. And when you open the abdomen, you see all those tumor deposits. But sometimes the disease is very advanced and you cannot operate on all of them. So you do chemotherapy first. But if you do chemotherapy, given that ovarian cancer responds very well to platinum-based chemotherapy, if you do the interval debulking surgery, you might not find residual lesions. And it's very hard to operate on residual lesions. So then we thought it would be interesting to devise molecular surgery, let's call it. And we know that ovarian cancer cells have folate receptor on them. And then we thought of doing a project in which we can devise a probe that can target our surgeon. So we had this project in collaboration with 2 other universities, where our main objective was to devise nanoprobes. So we have nanoprobes. We use ICG, and it's commonly used in various surgeries. Thank you. Thank you. So we use ICG, but ICG in itself is not very good for doing surgery because you have a peak of fluorescence only for a couple of minutes. So then we thought of making a polymer which could resist longer in the bloodstream, so the surgeon could see the tumors targeted by this compound, which will be linked to the folic acid receptor on cancer cells for up to 1 hour, and he could have the comfort of operating and seeing any residuals after chemotherapy. Of course, we couldn't deliver this onto real humans. We did it on mice. We are still doing this experiment, but we had some preliminary experiences only with ICG. So this is as you see, the operating field. This is how you see it with ICG. And we found all these black spots and these white spots. Black spot usually is fibrosis, and we put metal clips on the black spots and on the white spots. These are spots which still have vasculature remaining, and we send this for pathology to see how the intraoperative aspect would correlate with the final pathology. We also analyzed this folic acid receptor. We showed that it's very high before surgery. It's very high in advanced stage. We showed that if you do chemotherapy, it will decrease. And what was interesting when we analyzed samples was the fact that after surgery, in the first 3 days, there was a surge in the bloodstream of this folic acid receptor, probably by surgically manipulating the tumor. It is thrown in the bloodstream, but after 3 days, there was a substantial reduction in the folic acid from the bloodstream, in the folic acid receptor. Now, going on what we did on medical treatment. So I said we do neoadjuvant chemotherapy before surgery, and most of the literature says that it's okay if you do 3 to 4 neoadjuvant cycles of chemotherapy, but sometimes, given the clinical circumstances, and maybe we don't get a positive reduction after 3 cycles, we push another 3 cycles of chemotherapy before going to surgery. And this has been highly debated because they say if you do a lot of chemotherapy on a high tumor volume, you might impact tumor resistance because you are giving chemotherapy in the setting of a large volume disease. So the chance of obtaining cell resistance to chemotherapy might be higher. So we analyzed 25 patients who had interval debulking surgery and 64 who had delayed interval debulking surgery. And on our cohort, there was no evidence of acquired treatment disease. However, trials and retrospective analysis, multi-center retrospective analysis, are still trying to establish if this is really true or not. We also looked on the clinical features which are associated with BRCA mutations. Shortly, we showed that upfront knowledge of BRCA status is encouraged because we have this recent advent of targeted therapies, and we can make decisions when we know the BRCA status with much more confidence. For example, we showed that if you have a BRCA patient, BRCA positive patient, there is a higher chance for a complete pathological response, a higher chance for a platinum sensitive relapse, and there is a lower chance that your patient will change from platinum sensitive to platinum resistant disease. Part of my PhD, it's in ovarian cancer, the story of BRCA is even broader because BRCA basically means we have patients that are homologous recombination deficient. But the repair in ovarian cancer, the DNA repair, is basically divided into two main categories. You have those patients that are homologous recombination deficient, so their tumors cannot repair efficaciously the platinum-induced damage, and you have those that are homologously proficient, which know to repair their DNA damage. And those tumor cells are much more resistant to treatment, and those patients have a worse prognostic. And we tried to see how these markers, and it's a very complex machinery. We looked at a few immunohistochemical markers for homologous recombination, RAD51, ATM, FANCD2. We also looked for non-homologous end joining, which is 53BP1. We also looked at base excision repair pathways before chemotherapy, in red, in yellow post-chemotherapy, and at relapse. And there were quite interesting changes that we saw. And the long story short, these are also prognostic, most of them. And it's important to see how your tumor changes throughout the course of treatment. And from my point of view, it's more easier and more practical to do an immunohistochemical assay, because if you do a DNA damage assay, throughout the course of treatment, if you analyze DNA after chemotherapy, that DNA will still have that will be a DNA that will have scars, that will still have evidence of what happened at the beginning of the tumor, at diagnosis, not in the present. And it might not be informative. It would be like looking in the rear view mirror. So we think that immunohistochemical analysis would be much more practical. And finishing my presentation, we are also part of an international collaboration done with IQVIA, and it was very nice of them. They devised an R algorithm, which was sent to all study centers. We had a very well-defined study protocol. We extracted data from our patient registries, and we were able to analyze them locally. And after we had the local analysis, we sent centrally the results, which were blinded, and they were put together, and we got a very nice clinical description of cases. And this basically is a shortcut for GDPR. And it's very standardized because you have your database, you just install your R system, your algorithm, you upload your patient files, you analyze it, and it's just a click of a button and you send the data. This was my presentation. Thank you. Thank you.
Should there are some questions? I have a question regarding the quality of life on the
Yes.
So that was
So the study, the expression tree study, had close to 2,000 patients, but we extracted the data only for, we did like a subgroup analysis of the questionnaires from the Romanian patients.
And how many models did you test? The first one was the fit model, or do you understand? I used the structural equation model. That's why I'm asking you. It was fitting model from the first time, or did you make?
I think it was from the second time. We had an information that helped us with the structure. She had experience with the structural equation modeling, and she got a very good fit of the model.
Yes, I saw that. That's why I'm asking. Thanks.
It's funny, most interesting thing to me is the thing you said at the end, how to bypass the GDPR. Because we have a lot of problems.
Yes, you can elaborate a bit more on that. I would appreciate that. I was going to ask you that.
Thank you. So it was a study protocol, and this will be part. This was an ASCO poster. There were basically two ASCO posters last year in 2021 regarding this multi-site analysis, which were sent. And there's also the manuscript coming from this study. And IQVIA said that they will make publicly the script, the R script. It had a lot of technical challenges. Every comma, every punctuation meant your algorithm would stop. Sometimes it needed a few corrections, but in the end, the conclusion was that we had a protocol, we had a dummy database, we had very clear criteria on what to include, how to define lines of treatment, and we ran the algorithm locally in each center. It produced outputs, and those outputs were sent and they were analyzed like this in comparison. Of course, it was impossible to do inter-center analysis, but it gave an overview picture of the clinical course in ovarian cancer, and we had close to around 2,000 patients.
An AI method to anonymize the patients, if I understand right. Was that the main function of this?
Yes, so we did statistics locally in each center that was finally joined together. But given the fact that we used the same algorithm in every center, there were no variabilities between the statistical tests that were used. It was all pre-specified. All we had to do was to upload standardized data, to use standardized data to analyze it with the same algorithm in each center. And this is how you can increase the number of patients, because sometimes it's this problem of sending data to another center and you encounter a lot of issues regarding data protection. And this script will be publicly available.
Yes, one of the problem of rare tumors, especially gynecological tumor, is that at last in the period we study until 2007, the treatment and the diagnosis of patients were diffused in many hospitals. So there is no any kind of centralization, just it was very rare to centralize. Yes, but the majority were spread to many hospitals. And this, I think it's important to realize, to be conscious about this. And as I told you yesterday, and the country that where we studied is the question of volume hospital and the effect on outcome, were aware about this problem. But I think that it is the case to evaluate the evolution of treatment in hospital. So I mean the centralization in hospital where there are studies and collaboration with other centers. So
there were a lot of publications also regarding ovarian cancer surgery. Don't do it in small volume centers because patients will do worse. And it's proven in a couple of studies. They said you should have a minimum of 30 cases per year. Because it's very demanding surgery.
I have another question. We have heard from the lecturer from Nikolai before that actually they're population based and there are differences among different population and so on. And I could notice here that the survival, these patients at risk when they have this Yonsei, I guess that's South Korea.
Yes.
They have for everything higher numbers than the other populations in Europe, so that maybe a population difference somehow. Because it's a different population in South Korea and in Europe.
This is percentage of patients, and this is outcome of surgery. So this is number of complete resection they had. So it was around 60%.
No, in the lower table, at patients at risk, like survival, 1 year, 2 year, 5 years, you can see survival rates in the group of Yonsei is higher than every other group in Europe.
They have 24%. It's quite hard to comment because it's a very different population. What we also saw and couldn't find an explanation in this study, I didn't put here all the results, but they are in the poster and they will come in the manuscript, was the fact that patients from Yonsei and from Cluj were in average 10 years younger at diagnosis. So that was really striking, but we had a lot of patients. We had 400. We had a median age at diagnosis, I think around 54, and they had 63, 64 age at diagnosis, and we couldn't find an explanation for this. Maybe this would be also interesting to analyze. Maybe there's a mutation in the population that is responsible for younger age at diagnosis. Yes, in Cluj and in Yonsei in South Korea. It's not mentioned here, but we had younger patients. Sorry? It's Romania. It's true that we do a lot of surgery, and we also we have this gynecological department that does also benign surgeries, not only malignant. So a lot of cases are addressed to our institute. So this might be an explanation, maybe as a lead time bias. No, they received surgery and they also received chemotherapy, and some of them had neoadjuvant chemotherapy. And now we are working on doing something called Sankey plots, which is very interesting diagrams which are trying to explain how the population from one study center goes throughout its course of treatment, and it's quite hard to explain it without a graphical representation. So to see the flow of patients throughout treatment. Yes, but they were a minority. This is late stage and did not undergo treatment. And you see, it's with gray. It's very hard to see it, but it's this difference. So most of us, we had maybe 10% that did not undergo surgery, much more in Leeds, in Frankfurt, and in ICO. Thank you.
Maybe perhaps instead of considering only overall survival, we could consider something like age-adjusted survival or relative survival, or take into account the life tables of the particular population under study. Say, because as you're saying, the patients from Yonsei and Cluj were of a slightly younger age. So their life tables sort of would be, I think, in a different position. Maybe the more experienced statisticians here can sort of say whether it's yes. True. True. True.
Most of them were epithelial, so round.
Lots of confounding factors. Maybe correct for confounding factors. Quite complicated thing to do.
There is a very high interest in, from what we have noticed in the last years, for real world data to be produced and to be analyzed, because of course real world data has its disadvantages, but it's basically something that you are using in your clinic, and you can have a very large number of patients in which you can observe maybe some aspects that you would not see in a clinical trial.
This is one reason that you have to push for a national cancer registry information.
Yes.
Not only which is a very small population. It's important.
Yes, thank you.
Okay, so now we can go for lunch, and we have till, is it lunch? Yes, it's still 2:30. Yes, we have 1 hour. Yes, we can go for lunch now. We have 1 hour. Yes, and then we'll return, and it will be the talk of Dr. Vera Dimitrievska, and then of Dr. Federico Ortelli and Aldona from COST. Okay, so we'll continue discussing during lunch. Thank you. Vera's there. Okay, so we move on to the next part of our conference. We're nearing the final part in terms of presentations, and they will basically, following Vera's lecture, it will merge partly into the MC meeting, which then we'll see how it goes, whether we finish it before the coffee break or else we go by the plan, do the coffee break and then do the MC. We'll see how it goes. Okay. All right. So I'll introduce Dr. Vera Dimitrievska from North Macedonia. So Vera has a PhD at the University of the American College in Skopje in North Macedonia. She has 15 years of intensive and dynamic experience in international and in-country social health policy change projects, Horizon 2020. She has been managing professionally various projects, not only in North Macedonia, but even in the Netherlands, in Southeast Europe, including Serbia, Croatia, Greece, Bosnia-Herzegovina, and Kosovo, in government and non-governmental sectors. She's an experienced analyst, researcher, team leader, and networker. She's currently working in an EU regional project called B2Inf, which she will be talking to us about, to compare the citizens' participation and perceptions with information provided by assisted reproductive technology clinics to society. So thank you, Vera.
Thank you, Jean. I'm really glad that you sent me the invitation. So I'm scientific communication manager for the Cost Action. Along with Jean, we've been actually for 2 years and quite extensively collaborate. So when Jean sent me the invitation, I was quite surprised what to talk about, although I'm not a clinician. So then she just suggested, although she knows this project, to talk about this. I will focus more on the project that has started also before the pandemic, but then in conversation with Jean to make a link with this action. Mainly, I will introduce the project as an overview, what is the methodology, then how the data was collected, what are the main results, and in order to make the link with this action, I will just briefly present some raw data that I gathered yesterday for the rare gynec cancer and the main conclusions for the whole action and the project. So the project actually started, as I said, before the pandemic. It is B2INF project, which giving the voice to young population in several countries in Europe in order to see their perception regarding the ART, assisted reproduction technology for society. It's a 4-year project supported by European Commission, the Horizon 2020 program. The main aim, actually, it is related to know the perception, concerns, and expectations of young people from 18 to 30 years related to ART. And the target countries are Belgium, Switzerland, Italy, Spain, North Macedonia, Albania, Slovenia, and Kosovo. So this is the map of the project, how it's actually implementing still, and what are the main stakeholders in conducting the whole analysis and implementation. The main partners, as I said, are from those countries and mostly involves academia, but only from North Macedonia is one NGO because when the lead sent the invitation to be part of this project, we couldn't manage to include the academia, and then we decided to go with Health Grouper as an NGO. What is the actually justification, or what is the main reason why this project was initiated? It is actually considered that 186 million individuals suffer from infertility worldwide, which has a huge impact of the quality of life and the well-being. So that's why fertility care is considered to be an element to promote reproductive health. And 10% of the population born in Europe will be the result of the assisted reproductive technology, who want to have children in a different way. So in that way, ART plays a significant role in reproductive health of European society. And it tries this project to answer to 2 main research questions. How do young people perceive and think about ART? How can ART clinics better align their research services and information with the views, concerns, and expectations from the citizens? Mainly, the project is focused on qualitative approach with interviews and focus group discussions. It has 3 stages, and data is collected about perception awareness of ART among the young population. Then the data is now in the second phase, is analyzed from sociocultural, legal, and gender perspective. Currently, we are working on sociocultural perspective, and the 3rd stage is focused on dissemination of the findings through the distribution of guides with standardized guidelines. So the field work actually was conducted, last year from March to June 2021. 98 youngsters were interviewed, and 33 clinics who are doing ART through all these 8 countries, their sites were analyzed, which means we wanted to see how the clinics in Europe, in western part and southeast of Europe or the Balkan countries, actually present their clinics or sell the ART technology to the citizens. So average age of the participants was 24 years. And the interview guideline had 4 main topics: parenthood, infertility vis-a-vis fertility, ART knowledge, and publicity about ART. From those 98 interviews in the 8 countries, 3 teams actually emerged from the analysis of the interviews. Knowledge and perception about fertility vis-a-vis infertility, what are the young people perception on ART, and information and publicity about ART. Briefly, I will discuss the results, although there is still raw data and we are back and forth analyzing, presenting to a certain team, then scratch it and delete at some points that are not relevant or relevant. So, but I will just briefly focus on these main bullet points. That participants, young participants, described that parenthood as a relation beyond biological ties and infertility identified as a social taboo. This was actually very, very common for the southeast of Europe. Most participants perceived ART positively and would use it if it needed, although knowledge on ART among all young people is very low. During the interviews, because I was focused for the Balkan region or southeast of Europe region, they never heard about ART, but I'm pretty sure that there were some girls or women who had actually some rare cancer. Participants considered information on ART scarce and suggested that government-led information campaigns should be launched to raise awareness on it. And concerning the clinics, the websites provided information on infertility and exhaustive and technical description of ART is mainly that provided on the website. And it made difficult for the general population to understand, especially with the medical language. The data offered on success rate of ART was pretty much unclear among the participants that didn't have knowledge whether the ART should be successful or not, and heterogeneous. So to make the link with this action, we thought that with my colleagues that we can firstly see what is the data regarding rare cancer, gynecological cancer in North Macedonia. So in collaboration and with the help with my colleagues from the Institute of Oncology and Pathology in Skopje, yesterday we got very, very raw data from the these two type of cancers. So please take it in very carefully in consideration because it's absolute number. There is no still we don't know how it was collected. Still we don't know whether it's diversized. We don't have from the from the age perspective or from the region. So mainly the data is related to uterus, cervix uteri, and the corpus uteri. We didn't get the data from the ovaria or from the vulva, but for me, it is this absolute number from the 2019, 185 rare cancer data still is quite questionable, but still is also concerning. So we, as I said at the beginning, fertility issues among young women, females with GYNOCARE treatment should be linked with this action and the previous project that I'm working on. So what are their options if the women, young women from 18 to 30 are facing with the GYNOCARE, how they do the fertility preservation techniques? According to the literature and looking at the Google Scholars and certain papers that we work with Jean, there are two options for those women or the youngsters, clinical options and non-clinical options. I wouldn't focus on the clinical option because I'm not a clinician, but more or less, you know how that works. My colleagues, one of them actually work on that. So if he wants to, he can add something. But from the non-clinical options, the literature says or can use donor surrogacy with using different ART techniques and adoption when biological parenthood is not available. So that is general how we try to, with Jean, to link this project with the action. And that the main conclusion would be that reproductive health for females is very important for their well-being, reproductive counseling, post-treatment and pre-cancer treatment for those women who suffer from the rare cancer. And patients and their close family should be well informed about the availability of these treatments, non-clinical and clinical. And that in general, how we try to refocus a bit, because whole day we're talking about nanotechnology and yesterday different perspective for rare cancer. So a bit of light presentation from this point of view. So thank you, Jean, for the opportunity, and thank you to all.
I must admit that me, myself, being a gynecologist with a special interest in infertility, of course, I do see a lot of connection with women with cancer, especially rare gynecological cancer, which affects pediatrics and young women who might not have started their family yet or have not completed their family yet. Of course, knowing what lies ahead, like we heard yesterday from Sharon's presentation, that cancer is just a chapter in their life. It's not their life, it's just a chapter in their life. So it's very important that part of it is educating them about what would be available, and not educating them only, but educating the clinicians and the whole team that, you know, before a girl or a young woman starts her, I mean, life-saving oncological treatment, you keep fertility at the center of it.
Yeah.
In fact, I mean, again, I'm involved in other research work related to oncofertility, which has become huge. Oncofertility, I mean, with the advancement of cancer therapy and the increased survival in cancer therapy, of course, now women are not just surviving, but they're surviving and surviving well and starting families even after, or considering starting families even after having had a cancer. And although we know a lot about other types of cancer like breast and leukemia, lymphoma, etc., of course, I mean, less is known about gynec cancer, just because, of course, gynec cancer tends to be treated a little bit aggressively and even removing the reproductive organs. But of course, we need to start moving away from that, especially in young women who want to start a family after. So I don't know if there are any comments on this. Thank you. Thank you for helping us marry the two big projects. So thank you so much. And I think now I give the floor. Sorry, is there a question? Sorry, I thought we had a question. Okay, thank you. Thank you. Okay, so I think we can move on to our next presentations, which are going to be given jointly by Dr. Federico Ortelli and Aldona Gambalik, who are our two cost representatives from Brussels. So Federica, she is a PhD. Actually, her PhD is and postdoc is related to malaria vaccine development, but she has been involved in grant management and in the cost office since 2009. And she is the scientific officer in charge of our cost section. And then that closely flows also with Aldona, who is our administrative officer from the cost office. So, no, thank you.
Thank you, Jean, and thank you for the invitation. We are very happy to be here and meet the action a second time, so it's very nice. So when Jean invited us, she asked us to give a bit of an overview about cost and how cost actions work. So I will give that part, and Aldona will focus more on the funding mechanism. So what can be asked, what we support in financially with the cost actions. So cost, in fact, is an acronym, and we have lots of acronyms in cost. So it stands for European Cooperation in Science and Technology. And the purpose is to help pockets of excellence to collaborate at European level. And you will see we also go international and in space even with our action. These are the main features. So we don't have a work program like the Commission has. We are bottom up. So every year now we collect proposals. The next open call ends on 20th of October. So at the date we collect all the proposals, and then we start the evaluation process. And so it's really ideas that come from the scientific community. And these ideas are getting more and more interdisciplinary. So it's very nice to see that the combination of scientific fields that combine efforts to tackle a specific problem. And we also have different partners. So mainly we have academia in our actions, but we also have public organizations, industry, NGOs, sometimes WHO, and so we have representatives in the action, which is it makes it really interesting. We sometimes have policymakers, so actions manage to get the policymakers in from the beginning of the action. And it's really important if you want to have an impact at the end. In this case, for example, you have patients organizations and so on. So it's really good. We also have all career stages, and we have specific policies for young researchers. So they have a bit of a, we focus on them, we help them to emerge, let's say, in their scientific, with their scientific career. You will see we have COST member countries. So COST is not part of the European Commission. There is often a bit of a misunderstanding there. We are independent and we get funding from the Commission. So we have 40 members country. So it's pretty large. And near neighbor countries, I will show you the map, and then we go for really global cooperation. The strategic priorities, it's promoting and spreading excellence through networking and fostering interdisciplinary research, which is, I've seen the case these 2 days, and empowering and retaining, helping young researchers in their career. We currently have 292 running actions. So from all fields of science and technology, we have social sciences, also cultural heritage, astrophysics, and so on. And we are about to approve 70 new actions in a couple of days. So next week they will be published, and there are quite a few on cancer, genomics. So you should have a look at it and contact them if you find them interesting. So these are the average figures that we have, the number of training schools per year, the average budget, and short-term scientific missions, and the staggering amount of researchers that are involved in COST actions. So these are the members. We have 2 colors. The green ones, or yes, greenish, they are the so-called ITC, so Inclusiveness Target Countries, and this comes from the widening policy from the Commission. And then we have Israel, cooperating state, and we have since 1 or 2 years, I don't remember, South Africa as the partner member. So this means that when you have participants from South Africa, they get special funding to participate in COST activities. And our colleagues in the policy department, they are exploring other such collaborations with other international partner countries. So the family is getting bigger and bigger. And you might notice that we have Ukraine and Georgia now since 1 month or something like that. So it's good. So having all this geographical coverage, and now we have new rules as well at COST with the Horizon Europe. It means that you can involve researchers in your actions, whether or not you have an MC member in the action from that country. So if you have colleagues in a country, I don't know now, all the MC representatives, but let's say you don't have Portugal, for example, but you have a research group or researchers that are interested in participating in the action, they can do so. So this is new. Beforehand, you had to have an MC member representative. Now, anyone from these 40 countries can be in. Yeah? So that's important. And so now you see in green the near neighbor countries, and you see in orange the international partners, so the rest of the world, basically. And the numbers that you see here are the number of actions they are currently participating in. So Argentina is in 14 COST actions. Of course, the largest, well, Australia is pretty high, but I think US, yeah, 211. So it's quite an important collaboration there. So this is a yes, it's networking graph that shows with the links that can be built among different fields. of science and technology. This is an example, but from your action, I can already see a few bubbles connecting, and it's really interesting when we do this graph at the beginning of the action, and when we do it at the end, you'd be amazed. I will do it towards the end of the action, and I will send it to you. It's amazing to see the connections that are built, and it's really a field for opportunities. It's really nice. So the strategic priorities. So I will skip a bit the global networking and the science-informed policy advice, which are important. These are led by my colleagues in the policy department. Maybe more relevant for you are the Cost Connect. These are events that we organize at cost on specific topics that are usually in line with the priorities of the Commission. And we did one on cancer, how to beat cancer by 2030, Mission Impossible. That was the title. I don't know if you. And the purpose is to invite actions that work on that field, and also invite stakeholders, Commission, so that what we think are the main players in the field, and spend the day together thinking about the problem, how to solve the problem, what should be done, and so on. So we have quite a few coming up. You should check our website for further details. Then we have the Cost Academy, which is run by my colleague Anna Toivonen. And maybe Vera, you attended some on science communication. Yeah. And so it's science communication, leadership, sustainability of cost actions. We have one on how to engage with policymakers at European level. So this might be also interesting for you. So these are upon invitation as well. And but you surely have been contacted by my colleague. Action Chair Forum, yeah, that was a recent one, yeah. And now we started again on face-to-face at our premises in Brussels, so it's not virtual anymore. And then we have the Cost Innovators Grant. These are grants that are awarded to ending actions that have show an innovation potential. So when actions are ending, and this will be the case for yours in the coming months, we invite them to submit an application that has an innovation potential, and then it's of course evaluated. And if it's successful, we provide funding for an additional year up to 125,000 euros. But it's not for the whole action. It's a subset of the action. So it's a smaller team, and it's really dedicated to innovation. So I will be, or my colleagues, I don't know who will be taking over this project, will be inviting you in before the action ends. Yeah. So the impact, well, of course, scientific and societal impact through discoveries and breakthroughs, and this is maybe the most obvious one, but then also research career. So we see, or we get feedback from participants, especially young ones, that go for short-term missions to another lab. They get opportunities, they get career opportunities, they get published through the STSM. So it really has an impact on their career. And also the follow-up funding. This is very interesting. When you are in an action, especially towards the end, you have ideas that you submit maybe to the European Commission. Those proposals have a higher success rate than proposals submitted not within an action. And this is because you work together for 4 years. It's an interdisciplinary network. You get to exchange and understand each other. You tackle a problem from different aspects. And I think the success rate goes up to 35% for age 2020. Oh, 37%. So it's quite impressive. So I mentioned the scientific benefits to be in a cost action, but also networking, and I've seen it these 2 days. I think it's yes, it's almost superfluous to mention it. We just seen also today with the support the Bulgarian colleague received from the crowd. So it was heartwarming to see it, and wish you good luck and career opportunities and the financial support for further funding that emerge from the action. Here we have average values. We have 60 new actions. As I said, this time around it will be 70. Next time, so in October again, the plan is to fund 70 new networks. In the past, we funded 40, so it's quite a big increase for us. These are the spin-off funding per action on average, 5.8 million that the Actions Network get after the action. And this is across the horizon 2020. So this is data that is retrieved by my colleague at Costa. So these are the news, the 70 new actions, this I mentioned, Ukraine and Georgia, they are in the COST family now. Okay, annotated rules, that's very specific. The COST Academy I mentioned, and this is, I think, very interesting for you, because recently the executive board of the COST Association analyzed the impact of the pandemic on actions of the cohort, so funded in 2017 and 18. And they listened to the scientific community as well and decided to grant, if you wish, an extension of 6 months based on force majeure. So it's 6 months with additional budget. It's not only in length, it's also financial support. So this might be an interesting. It's we don't have it yet. It's going to be calculated based on the countries that are in the working groups. So you should populate the working groups. So the cost actions, that's what we found. So they last 4 years. So you're about to end, well, in March. So you still have some time. The initial number of countries of these 40 is 70, but we have actions that reach 30 countries with MC participation. Then you have to have research, coordination, and capacity building activities, and the average budget over the 4 years is 600,000 euros for these networking activities. So we do not fund research in itself. We fund the networking, and what does it mean? We fund meetings like this one, and in the meetings we reimburse the travel and the accommodation. And Aldona will be much more eloquent than me about all the possibilities. So maybe I just for each of these explain what is meant with it. So we fund internal meetings like management committee meetings and core group meetings. The training schools, these are typical training schools or students and trainers. Usually we have 1 trainer for 3 trainees. And usually training schools last, I think on average 3 days, but they can go up to 5. Aldona will explain what we fund there. Then we have ITC conference grants for young researchers. And young researchers, we changed the definition with Horizon Europe. It's researcher that is 40, younger than 40. And these are exclusively for researchers affiliated to an institution based in this inclusiveness target countries that we've seen before. And they cover, so the purpose of this is for this young researcher to go to an international conference, so not organized by the Action, and present a poster or even a presentation. So it gives them the opportunity to be exposed, let's say, to an international environment. The short-term scientific missions are also very, very popular, and they allow for researchers to visit a lab in another country. So it's not within a country, you have to go to another country. And they're very popular because you can, for example, as a young researcher or as a researcher, go to another lab and use sometimes lab or learn techniques that you don't have within your country or easily accessible. So this gives really nice opportunities for researchers. Then we also support dissemination and communication products. So the Action website, publications, flyers, videos, guidelines, so on. The virtual networking grants, this Jean mentioned it earlier on, they were introduced because of the pandemic, because of course funding networks and collaborations and travel, it was a bit constraining for the Action. So this was a way to allow researchers to still exchange and having some financial expenses and gains. The dissemination conference grant, this is for in DMC for someone to go to an international conference again and only oral presentation about the Action, of course. So it's opportunities to go and everywhere in the world to give a talk about the Action. Then other expenses, these are typically bank charges as well, but I don't want to steal Aldona's points. And then workshop and conferences like this one, and working group meetings, of course, to coordinate the research within the Action. So I think, Aldona, maybe we take the questions afterwards. Sorry.
Hello to everyone. I'm also very happy to be here. It's the first time for me to attend an Action meeting, so I'm kind of stressed. I hope I will explain you well everything, but otherwise we can still be in contact afterwards. So who is eligible to receive the funding from COST? Everyone is eligible to participate in COST Action networking activities, but not everyone is eligible to receive funding. That's I hope is clear. So as you can see, it's mostly the COST members who are eligible, near neighbor countries, organizations, and for training schools and meetings. Only one in Action lifetime, the IPC and the international organizations can attend. Yeah, how the funding works. So we found by grants, by travel reimbursement, and invoices, and I will go into details later. So I will start with travel reimbursement rules. So it will apply all of you, like for today, you came for a meeting and you will claim the costs for your long distance travel, which is above 101 kilometers one way. And you will also receive the daily allowance reimbursement. So daily allowance covers your accommodation, hotel, and meals. And as well the short distance travel, which is the travel from the airport to the venue, or for from your home to the airport. Yeah, it's all travels below 101 kilometers one way. So the daily allowance is based on the meeting country. So there are different rates for each country. where the activity is taking place. I think for Malta, unless you didn't set your own daily allowance, because this is also possible, it's 193 euros. So for each full day, you will receive that amount. And then based on the travel times, depending on what time you left your home, what time you arrived to your home or affiliation, you will also receive a percentage of daily allowance. Then for the long distance travel, yeah, let me just mention also that for daily allowance, you don't need to provide any invoice. So no invoices, this is kind of one lump sum. And for the long distance travel, you can travel by train, by plane, even ferry, and by car. For that expense, we require the invoice, which mention your name, your place, what time you left, you took a train or plane, and the amount you paid, so the invoice. For the car, it's limited to 2,000 kilometers and calculated 0.35 euro per kilometer. And we have also other costs you can claim. It's mostly visa costs and travel cancellation insurance. Just to mention here, only travel cancellation insurance is eligible. No travel medical insurance or luggage insurance. This is not eligible. So important to remember. I hope you already did it. So before the event, once you receive the invitation for a cost activity, a networking activity, you have to accept or decline the invitation in eCost, update your bank details, and you can also through the eCost calculate, estimate your expense, which you can reimburse after. Sorry, no, not yet. And during the event, you also heard from Jean, it's very important to sign the list each day. Thanks to that, you will receive the reimbursement of your travel. And after the event, you have 15 days to submit all necessary documents and your claim. Then for the grant holder, it will be, I guess, Diandra or Jean. She has 30 days to proceed your claim. So in total, it takes 45 days, takes 45 days. Sometimes there are some delays, I guess, but yeah. So for that, it's all important to know non-eligible expenses. So it's everything which goes outside of the long distance travel and daily allowance. So registration fees, fuel, parking fees, taxi, this is all not eligible. Taxes and other additional accommodation expenses. And now to mention about local organizer support. So local organizer support goes with meetings and training schools. So any eligible local entity can apply to be a local organizer. So for this meeting, it's Jean, her institution, who applied to organize a meeting and to host you. So any of you who are a full member country can also apply to be a local organizer. For such a so local organizer support, it takes a form of a grant. So it depends on few parameters. It's a format of the meeting, so it can be face-to-face, it can be hybrid, like is the case of the meeting, today's meeting, and also virtual meeting. For each of them, we have a specific list mentioned in details in the annotated rules, what can be covered within the grant. For the grant, we don't require any invoice, so we will not ask you to provide the proof for each expense you did. But in case of any audit of your institution, you always have to prove the expenses incurred. So also the calculation of the grants depends on the unique attendees, the number of unique attendees. So to give you an example, you had a meeting of today's meeting, and only one participant attended both days, but for the calculation of LOS, he will be counted only once. So it's a unique participation. And also, well, the duration of the meeting. So in our annotated rules, you have all details and the maximum amount you can claim for the meeting or training school. To give you an example, what can be covered is the renting of the room, conference venue, refreshments, coffee breaks, yeah, the administrative support. This also can be covered. Important it's to send the ECOS invitations to everyone, also for those who are not claiming the reimbursement of travel. So you have more participants to calculate for LOS, and that everyone signs the list. That's very important. For the virtual meetings, we require an attendance log. For instance, Zoom or Teams, they provide such a report, and this has to be included in the LOS claim. For the grants, so we also distribute grants for short-term scientific missions, ITC conference grants, virtual mobility grant, and dissemination conference. For STSM, so as Federica explained, this is to host organization located in different country than the country of affiliation. So researcher can receive a grant up to 4,000 euro, which is paid after the receiving the report and approval of report. So after the STSM, participant can apply for a kind of prepayment, but this is up to the grant holder institution, because it's kind of a risk. It's not anymore possible to encode it in eCost. So we do not encourage a grant holder to do it, but it's as I say on the risk of institution. Then we have the ITC conference and the dissemination grant. So for that one, participant can receive up to 2,000 euro or for the face-to-face conference, or participate in the virtual settings, and this is up to 500 euro, also paid after the approval of reports. For the virtual mobility grant, it's a bit less, it's 1,500, the same after the report, and all grants must take place during the grant period. So this is also important. They cannot overlap between two grant periods. Mm-hmm. For the dissemination communication products, here we require invoices. So for each product, your action is requesting funding. You have to provide an invoice. What's important to mention here, that you can cover the scientific publications, open access, action website and the logo, multimedia, and also something new in this new grant period. It's booth at the conference. So this is, I think, very interesting for you. Like you can attend a conference and present, show your action to the wider public. Mm-hmm. Yes, I have prepared also a small update about your action. So just to mention that that you started this grant period. It's a third one, and you started the beginning of November. Will end in the 31st of October. So important to know and to remember to not organize any activity 2 weeks before the end of the grant period. Otherwise, it creates a bit more work for a grant holder, institution, and action chair. Mm-hmm. Mm-hmm. Yes. Mm-hmm. Yeah, it also depends on the cost. Maybe Federica can help me in that, but I think. Mm-hmm. 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 Yeah, and then also it depends on the like you have to sign the AGA, and maybe this takes sometimes a bit more time that not everyone is available to sign the contract directly. So it may be a slight delay, but normally beginning of November it should be already done. So for this grant period, Action received 159,000 and 499 euro, more or less. This is including SAC. And there will be no carryover. So we encourage your Action, to spend really all budget in this grant period because it cannot be transferred. And the next grant period, this Federica will maybe explain later to during the MC meeting, but it will depend on the network size and then the Action performance. And about the payment of the Action grant budget, so the beginning of the grant period, once the Action signs the agreement, the Action received the 50%, or this is a first installment. And then depending on the expenditures, if there is a need, the Action can apply for the second installment, which is 35% of the total budget. And for that, the Action needs to apply with the intermediate financial report. And the third installment, if needed, it's at the final etap with the final financial report. This is an overview of your of the grant period 3, but I don't know, Jean, maybe we can go into details during MC meeting, because it's just to see how much it's actually from the working budget plan. What did you plan, and mm-hmm. Mm-hmm. Yeah, it's just some kind of overview. So of course it can change, and you can reallocate the grant budget which you specified for instance. I remember there was an issue with the working budget plan. You couldn't include the virtual networking support grant. That's why here we have 0 in virtual mobility grant and VNS. but it was included in the OERSA, so other expenses related to science administration. That's why it's 19,000 here, but this will cover the 3,000 and 4,000 forecasts for the virtual, I don't know how to use it. Yeah, so it's this 3,400 which you plan to spend. Yes, something important to just mention here, that you should always communicate cost with the MC decisions and action results. And when you are writing an email to me on Federica, to not forget to mention your action number, because we receive a huge amount of emails, so sometimes it's difficult to remember the action number. And that's it. Thank you very much. And if you have any questions, we are free to answer with Federica. And thank you.
So you were very good. I mean, it didn't show that
Thank
was your
you.
first. And we're honored that you this was the first one, because you always remember it now. Okay.
That's very nice. Thank you.
So I have some questions. So first of all, so we've got one partner from the US. So being from the USA, would I mean anyone from the USA, if they were to come to a meeting physically, would they be able to be reimbursed, or?
Yes, so this is considered as a third state country, or IPC. There is I don't have it on the presentation, but there is a limit in attendance. So in the annotated rules, there's this big table which shows well, and I guess for participation in a meeting, it's once per action live time.
So they can actually be invited, for example, for a training school.
Exactly.
Or for one meeting.
Exactly.
But it's once in a, okay.
For training schools, yeah, the trainers can come more than one, so this is okay. It's only for meetings. So we have really specific categories for each meeting, and you can see for those kind of participations, there is a limit, but
it is possible that
yes, there is a possibility.
It was good to know, because I mean, now that AFSHIN has applied as part of the USA, so if there are partners in the USA, it doesn't have, I mean, it could be AFSHIN, it could be someone else, but I know that David is connected to the USA. So if, I mean, you deem fit that we invite someone over, both as a trainer or as a participant, it is possible, and there would be funding for them, which is important.
Yes. So for instance, here you can see for meetings, there is this IPC and international organization. This is only once per action lifetime for the meetings. And for training schools, it's only as a trainer.
Only as a trainer.
So if you want to invite a trainer from US, you can do it.
And the trainer would be entitled to reimbursement, like travel and the daily allowance.
Yes.
That's good to know. With a meeting, it could, yes, it would be covered. Exactly. Very good. And the question about November, so that's sorted as well. So it could be possible to combine it with a meeting in North Macedonia. I also had some questions about virtual mobility, because for some of the collaborations, there might not be actual physical travel. For example, just to give an example, like if someone wants to collaborate, for example, with David as a biostatistician, and they don't need to travel physically to Israel, but they can collaborate virtually, and they apply for virtual mobility. If you could just elaborate a bit how that works. So, I mean, I know a bit about it, but maybe for the sake of the for the members, just what needs to be done, basically.
For the grant, so there is mostly a report to submit about the work, and I have no example, Federica, if you remember any good example to give about the.
I can tell you what I did in the previous one. So in the previous grant period, there were 6 virtual mobilities. So of which I know there was Bridget, there was Miriam, and there was Olga, Sharon, Rita, and Ricardo, exactly. And at that time, I mean, unless the rules have changed, but at the time, they needed to submit an application, which is an online application. And then there would be the time period, which obviously must not be
overlapping the grant periods.
Over October. So it has to end definitely latest by mid-October. And then once you complete that, there is a report saying